Good. One more time. My name is Seung-Gyu Lee, Vice President, Korea Bio-Industrial Organization. Thank you. It's my great honor to welcome everyone to 2023 Global IR Teaching at JPN. On behalf of three co-organizations, Sidley and BioCentury. Thank you. And I also give my special thanks to Joshua Hopheim of Sidley of BioCentury. This very unique style IL event was created to increase investment opportunities and to raise awareness of the importance of bio-startup acceleration within the bio-industry ecosystem, especially during this week of JP Morgan Healthcare Conference. I am going to hold the event on site after 2 years of virtual demo days. Through this event, we hope to see more innovation in the industry by the bio-startups, and we believe this global IR teaching event is a significant opportunity not only for venture capital investors and bio-startups, but for the entire bio industry. I would like to thank you, all the investors here to share their insight with fabulous 12 bio-startups and our co-host, Sidley and BioCentury. And not to forget, NASDAQ for sponsoring such a meaningful venue, especially at the very valuable time during the JP Morgan Healthcare Conference. Thank you. And I hope you all enjoyed the Global IR and JPM. Next, now we will have Joshua Hopheim, co-head of Global Life Science, for one of our co-host, Sidley Austin. Thank you.
I won't keep you all because I know I'm not who you're here to hear from. But just wanted to thank everyone for joining us today. I'll give a little bit of background on Sidley Austin when we do a fireside chat and a case study on a deal that we did a couple of years ago with a Korean life sciences company. But just want to say thank you to the staff from KoreaBio and the support from BioCentury for this event. It is a fantastic day. There have been some amazing companies that have come out of this, several that we represent, fortunately, and I'm pleased to share the afternoon with you. David, if you could come up for just a moment.
Thank you. And can you hear? Okay. So on behalf of my colleagues here today, Josh Berlin and someone you'll be seeing a lot of very shortly, Jeff Cranmer, and the rest of my colleagues at BioCentury, I want to thank everybody here for including us here. I do need to say I'm obligated by my staff and my co-founder of BioCentury to tell you this, that tomorrow will be the day, the 30th anniversary of when we launched BioCentury at H&Q. And so thank you. So we've seen a lot of H&Qs, and one of the things I will say is that over the last few years, one thing that's been really palpable and noticeable is when the Korean delegation creates an event, good people, quality people show up, which shows the interest that you all are creating, not only here but elsewhere. But the point is, it's the trajectory is the right trajectory, and we're happy to be part of this today. Thank you.
So thank you, Dr. Lee, and Josh and David, and all our co-organizers, Sidley Austin, BioCentury, and of course NASDAQ for sponsoring the event. because we have such a good venue here. And I'd also like to give a big shout out to all the audience here today. It was nasty weather, but it stopped. It's obviously sunny. My name is Juri Huang. I'm from Korea Biotechnology Industry Organization, who initially designed this event back in 2020. We sat down with BioCentury and Sidley together to, make more cross-border investments happen. And unfortunately, throughout the years 2020 and 2021, we held an event online, and we're very excited to be here. This is our actually first in-person event, but our third event. We're very excited to be here in San Francisco at the JPM Week. Today, especially with the pandemic narrative, the macroeconomics did not seem quite really likely to us, and with bad inflation, and especially coming from Korea, I can say that the last 1 or 2 years has not been one of the heydays for bioindustry in Korea. So I just wanted to emphasize today, more because of this kind of environment, we definitely see a more ample opportunity for a rebound in the industry, which means more cross-border investments happening and deals. In other words, the show must go on, and I believe that's why we're all here. So with the full hopes, we have invited the 12 aspiring biotechs, 6 from Korea and 6 from global, and the time slot they're given will be 6 minutes of pitching and 4 minutes of Q&A with the VC panels. And we not only prepared the company pitching today, but we have prepared a fireside chat, and in between, we'll have case study sessions. So for the part 1 fireside chat, as we move these great panels to the podium, we invited one of the most active healthcare investors from US and Korea to talk about the outlooks of 2023 global healthcare industry, chaired by Jeff Cranmer, who is currently the executive director and head of daily intelligence at BioCentury. So it's on you, Jeff.
Here.
I guess I get this one. All right, good afternoon, everyone. Thanks for coming out, dodging the raindrops, to join us here this afternoon. I'd like to welcome our 3 excellent panelists back again this year. They were with us last year virtually. And given that we have a short leash, I'd like to ask our panelists to quickly introduce themselves before we dive into our conversation. Michael?
So great to be here, especially my former hometown of San Francisco, and also my hometown folks from South Korea, since I was born in Korea. I work at CBC Group. We're a healthcare dedicated investment firm. We manage over $7 billion today. Hopefully by this end of this year, we'll get to about $9 billion. We invest across strategies in equity, private and public, as well as credit, and we invest across therapeutic diagnostic devices. And we recently did a big deal in South Korea, and we hope to do more. And we have a background of physician scientist trained here in the US, being a venture capitalist on public investing, and I was once a public companies here in South Korea. I'm based in New York, so it's great to be here with you guys. Thank you.
This is Jiu from Daol Ventures, but I used to be, our company's previous name is KTB, so everyone knows KTB is the first Korean VC since 1981, and we are the first Korean cross-border VC and opened office in Bay Area in 1988, and then opened the first Shanghai office in fact early 2000. So I'm charging of healthcare investment in our group, and then so we are not actually life science focused investment firm, but one of the major sector was healthcare, so I'm charging of that.
Great. Thank you.
All right. Yeah, it's great to be here. It's a sunny day right now, but it's been rainy. So my name is Min. I'm a managing director at KB Investment. So KB Investment is the 100% subsidiary of KB Financial Group, which is the largest private bank in South Korea. We were founded in 1990 with the asset under management about $3 billion, only in the investment side. And we're one of the top, investment firm in terms of biotech. Not only we invest in biotech, but also in a variety of sectors. Also in biotech in Korea, but also in global side. So, happy to share some of the experience.
Excellent. Well, gentlemen, stormy skies to welcome us into BioPharma's 2023 kickoff event in year 3 now of this biotech bear market, but there's sun out, and I've heard a report of a rainbow from one of our panelists, so hopefully that bodes well for all of our presenting companies going into this year. Now that I've set the mood, Min, I'd like to start with you. How are you thinking about your investment strategy, and what's your outlook for the year ahead?
Sure. So, it's always time of the year that we share the investment strategy, but I'm going to share more into not with the investment strategy, but the companies that I really look for this year. So if you do the analysis last year, probably due to COVID, if you see the companies, for example, pharmaceutical companies, they have gained much of cash last year, probably like 10% for some of the company. I've seen about 22% for Pfizer, for example, probably due to COVID with a cash projection. And then, with that being said, there's going to be a lot of chance, for those biotechs, potential to be merged and acquired, or research collaboration or strategic investment whatsoever. On top of that, you'll see some of the blockbuster drug, their patent being, expired soon, within 1 or 3 years. So they are going to look for, I know there's a lot of pharma companies, it's really weird to say on behalf of them, but I'm sure there's going to be a lot of opportunities for the biotechs. Second, the market IPO market is being very smart these days. But the people ask me, what do I need to do? The answer that I give to the people is just focus on the fundamentals, what you're supposed to do. Being fundamental means, not the potential. You don't want to have the value driver because of the potential. You want the value driver from the data and evidence of the science. So that's what you're supposed to do for the biotech. So you want to show those kind of stuff. And third, you need alternative value driving factors. So if you have a platform technology, so in other words, it's going to be a plan B for the investors and public. So you need some kind of comfort to the market that you have a second asset, third asset ready to go. So platform technology will be one of the sectors that I will be interested in for year 2023. Yeah.
Yuhan?
Yeah. So like everyone knows, and the words cannot explain how tough last year is. But I do think that this year would be almost same. But it would be a top years again. And then I always told my portfolio companies, management team, that just prepare for the worst scenario cases for this year as well. But there is another point is that actually, even myself, I cannot invest many deals, many company, and then also very smaller money than comparing with the other years, maybe 2021 or 2020. But that means there is some dry powder in our investment firm. So the number of deal will be increased, but the average size will be smaller. So and my strategy on this year is that focusing on try to find the small and smart deals. So that's my strategy, and then maybe more conservative thinking is in the management team in investment firm. So you guys should consider about that, and then try to meet the investment candidate as many as possible. So yeah.
Excellent, thanks. Michael?
Sure, I'd love to be contrarian, but I won't do that right now. But I have to say, obviously, if you've been doing healthcare investing, been in the industry long enough, you've seen through different cycles, from the dot-com 2000 to the 2008, and even a few years ago. So it's not surprising we're going through another cycle. Healthcare is defensive to a limit. And I think we've been a beneficiary of easy capital for some time, especially in Asia. You could talk about China, how we never had a down round in China for 7 years. There's only been a bull market. In Korea, same way. There's been a lot of easy money and easy way to make money as an entrepreneur. Now we just have to be efficient and realistic. So I think 2020 was great for our firm in terms of doubling in our size, in terms of the money we managing, because our portfolio company has done really well. So we've been a beneficiary, and a lot of companies raised a lot more capital than ever before. So that's the positive. But last year we went through some correction, but sometimes there's a healthy correction you need to go through. So it's like pruning, right? You can't have easy money forever. So I'll say, you know, you asked me a few months ago, before all these trade wars and the macro and the zero COVID policy was still fully intact, maybe I was a little more pessimistic. But with the new year, with the sunshine and the rainbow, as you said, and you asked me to be positive, so I will be positive. I am
I had to
optimistic.
bribe him a
cautiously.
little bit on that.
Yeah. I'm cautiously optimistic of what's to come. But I think you could talk to entrepreneurs. You just have to be creative, patient. It's not going to be, you know, oversubscribed round with easy step-ups. But that doesn't mean you can't be creative. And what I mean by that, there's access to equity capital. There's also access to non-dilutive capital, and maybe partnership you may have to do early. So there's other ways to get capital. So I'm optimistic in the sense that, especially for us with new capital to invest in new companies, is a great time to invest. For our portfolio companies, we invested before valuation went up high, a little bit difficult. But that being said, since it's a new opportunity, I'm very optimistic and happy to meet you today.
And how are you dealing with that situation?
In terms of?
Well, the mismatch in valuations that you just raised.
Yeah, so, you know, we have difficult board meetings in terms of with our private companies. We certainly have thought about, you know, asking the companies to conserve cash. We definitely been in a buying spree, as you know, with our fund. We've done a lot of in-licensings, and we bought a lot of assets, paid a lot of money, and now you have to say, realistically, easy money is not there. So we do have to make difficult decisions at the portfolio company. We review a lot of the R&D pipelines, prioritize. We certainly have cut costs by letting folks go. You have to make difficult decisions because you got to conserve cash for the long term, survival of the company. So certainly we have made difficult decisions in the last 12 months. On our equity fund size, we haven't really invested the last 6 months. The reason is because we know if we wait a little bit more, valuation will come down. But now we're ready, right? Rolling up our sleeve, doing more work, because I think bottom is not necessary here today. It may be for another 6 months, but you got to start the work today. So, you know, we remain optimistic. Our P fund, we still have about $700 million left. We raised a new venture fund. We have a new royalty credit fund. We've got a lot of dry powder to invest, so we're pretty excited.
Excellent. Min, I'd like to turn to you and springboard off that. Given the state of the capital markets as we've been discussing, how are you advising your portfolio companies to survive this?
So, yeah, just like Michael have said, I'm advising companies to preserve cash as much as possible. You know, it's just, you know, preserving cash, it doesn't really necessarily mean that, you know, you're just making a survival runway. It means more than that. You could be more creative in terms of business development, in terms of, so you're going to be the first candidate to be reviewed, probably by the other strategic investors or other firms for reverse merger and acquisition, for example. I'm just giving you an example. And there's all kinds of you know, flexibility and creativity that you could deal with if you have a lot of cash. The way to do it, there's so many ways. Unfortunately, some of the you know, cutting down people could be inevitable. However, that could be done. Extend payment schedule for some of the vendors, that could be possible. But not only just preserving cash and, you know, saving this cost, I would also want to emphasize some of the additional funding strategies. So I mentioned about the you know, increase in cash projection for the, you know, pharmaceutical companies. Now that could serve as one of the strategic investment for those pharmaceutical companies. And these days, surprisingly, well, not surprisingly, I know that there has been so many government grants that the government is supporting Korean companies or any companies in the U.S. too. So just utilize those kind of various types of funding that will be helpful for the companies, and that's how I advise usually to the companies to survive.
And Ji-Yoon, what are you advising your companies?
Actually, all of my comments, which I already prepared, are actually the overlap by the mean. So thank you. Yeah, I have the same idea, but more specifically, and maybe I just want to share two cases
Okay.
And one case is that we can see that there would be the higher interest rate from FRB, and then it will affect all over the world. And then it is directly affect the venture market as well, because some of LPs are not happy about some of LPs happy about the high interest rate. For example, in bank or national bond, something. So it will directly affect the alternate investment, such as venture capital. So we can see that there would be some very downtime for the investment. So I ask all of my portfolio company to cutting down the operation cost. So every single penny would be very, very important. But there is one company, and they reject my suggestion, and then they said, we actually have a calculation, and then only 3 weeks we can actually extend our runway. But that 3 weeks actually became very, very crucial, because it was last December. So in December, everyone go to some holiday. So he actually came to me and then give us his regret that, hey, Jun, I should have listened to your advice, because these 3 weeks, he didn't expect these 3 weeks can be very crucial for the company. So in that case, please save every single penny. And the other case is that, so maybe from after Corona pandemic, there were just so many monkey tests, some requests from all over the world, and we are maybe shorting out about that the monkey test, because mainland China lockdown. So I try to utilize my own network to introduce some of the hidden CRO in Korea, in Taiwan, and Japan, and they at least finally they find it. So, but they're very surprised that it was just the 50% compared with the CROs in the United States. So after that, they try to find those kind of a hidden CRO in the world. So crossover investor like me, and maybe I can be very helpful for that, because I get a survey together with my previous colleague in Japan and Taiwan and China. So you need to use a more smarter way to find some, not only focusing on some Charles River or something, but in this case, you have to save the money, so try to find a smarter approach to even in CRO.
All right, so sound advice for preparing for surviving the bear market and for life beyond. Well, I wanted to pick up a little bit from a theme that we touched on last year. What do biotechs, in particular Korean biotechs, need to do to make themselves attractive to global investors such as yourselves, especially once the market turns around? Michael?
That's a great question. I think I made some comments last year. I think one of the reasons a lot of Korean companies have gone to clinical stage quickly is because there's a dearth of capital if you're preclinical. There's a lot of valuation and venture capital investors in Korea. They'll give capital if you went into clinic. And I think with that, sometimes they went to the clinic a little bit too early without knowing a lot about the molecule itself, as well as maybe that's not the best indication to go after. So with retooling that's happening today, I would say companies should really look at carefully the indication they went after. And as you said, last 2 years was difficult in Korea because some of the more mature Korean biotechs have been around for a long time, going after indications that's probably not worthwhile. Went into phase 3, some of them had a spectacular failures, some of them didn't have partnership because it wasn't the right play. So I think one of the things that's really important for biotech is you have to decide to make a pivot quickly. You could reinvent yourself. You could have 9 lives. It's okay. There's enough capital. So if the original business plan doesn't work out, give up. It's okay to pivot to new things. So right now, I think you could use many different excuses, such as limited capital. The old investors don't want us to pursue new indications, but now I have new investors that want us to go a different way, that's sexier and more commercially viable. Make that pivot. Sometimes it is difficult, especially when you have a founder culture that doesn't want to go outside of that person's expertise. But in order to survive, get the U.S. investors. U.S. investors are used to recapping and backing the, failed in one clinical indication, but we're okay for the next one. right? And there could be a value that could be created. So that change of mentality or the old business plan to new has to happen. Last year, I talked a lot about CMC and whatnot because that's some of the challenges in the biologic space that a lot of Korean engineers don't have expertise. There's nothing you could do. If you don't have the experience, you can't make it up. But you learn as you go, and that's okay. And even for a lot of the public companies in Korea that fail, they got to make the pivot quickly. Then I think new investors will be okay with that failure because you're going to learn and make that indication change. So that's what I would say. That's really important. I think a lot of investors have, when they looked at Korean companies, especially last 10 years, have said Korean companies more advanced than a lot of the Chinese companies, probably because a lot of the Korean companies are already in the clinic, at clinical data. That's where there was a lot of in-licensing out of Korea. Of course, last 5 to 7 years, China just bloomed with new innovation and the discovery that's trying to catch up. But historically speaking, there's a lot of experienced entrepreneurs in Korea. So I think there's a way to redo this. So that's something that we, if we could have a conversation, we could help think through and make the new pivot that's necessary.
Excellent. June?
Yeah, so, but I don't want to touch on the science or fundamental research, but because it was no doubt it. The biggest difference compared with the Korean biotech and then biotech in the United States is how do they operate or how they make some organization by, some decision by decision. So recently I tried to tell my portfolio company in Korea and then tried to set up the frequent board meeting, and then you please use a board member to decide together. So previously, usually as the one CEO or largest shareholder founder, they decide by themselves. The decision would be very, very slowly. But so my advice to them is that to make a board members and the board meeting very formally, and also make some of a committee, like compensation committee or some of a committee, and then if so, maybe it looks like a more transparently managed by some board member or some third party committee member and join. So this kind of some thing is which the Korean biotech should learn.
Min, what advice do you have?
Yeah, so actually, with your question, I know you mentioned about after the bearish market, but if you look at retrospectively, if you talk to anyone last year, for example, the market was bad, and when they ask you today, probably they say market is still bad, and when they ask you tomorrow, it's going to be market will be bad. But with that being said, I'm not going to be saying what you need to do after this bearish market. I'm just going to tell you what are some of the common traits or common characteristics that companies have successfully been funded recently. So I'm going to just emphasize on 4 things. One is they have been focusing on the fundamental. I'm emphasizing this enough because many companies they tend to be overvalued because of their potential. I want to see the companies who have the right valuation with the right data with the right evidence. Second is it's if you're a pilot, you just go into the smooth air. It's easy to fly, but it's the skill of the pilot that actually distinguishes when there's a turbulence. Well, landing and takeoff is the actual thing, but I would say turbulence. So just, be it, ability to kind of go through the turbulence, meaning this bearish market, it's going to be more important, how to do a flexible business, creative thinking in terms of business development. Number 3 is the communication, whether it's to the public, to the investors, talk to them often. Don't afraid of, presenting bad data. Just talk to them and get the solutions. Just be interactive. And number 4 is the selecting the right investors. Don't just go after the one-time money. I guess you have to select. The biotechs need to select right capital who can make a follow-on investment. Also, who shares your vision and thoughts. So those are some of the common traits that have successfully been funded recently, and I think this kind of trait and characteristic will continue.
Excellent.
Maybe
Sound
one,
advice.
one other thing you mentioned is the valuation. I think
Yeah.
Korean companies had a higher valuation than a lot of U.S. companies. And
Yeah.
last few years, Chinese company had even higher than U.S. companies. Now I think it's all equalizing. Like Hong Kong Exchange is no longer premium to NASDAQ. Maybe it's discount. And same thing, Korean Kosdaq maybe was premium for phase 3 asset, right? They're multi-billion. So I think that being equalized should allow entrepreneurs to think, if you want global investors, you might not get the same valuation as you would get from Korean investors. So I think, one of the things we talked about, there was an event for the Chinese companies over the weekend. Same thing before, a lot of Chinese companies are getting local investors in China, easy money, to step up valuation now, because a lot of the global guys are not investing. Then it makes it difficult for the next round for us to go in, because you had the artificially inflated, who wants the down round? Entrepreneurs get killed first. So that's why don't take easy money. If you want a global investor, be reasonable with valuation, try to get the investors to come in earlier, because we'll never invest in a company in Korea with 600 million valuation, that's phase 1.
Yeah.
It's just difficult, right? So I think that's the reality. And if you need to recap, then you just have to think about spinning out assets and company and get funding that way. But we like good value nowadays, right? Because public comps have come down. For private companies to get capital from global companies, you got to have valuation that's akin to the U.S. level.
Excellent. Well, I think I have time for one more question. So maybe try to end on the rainbow note, the sun and rainbow note. What's one thing that has you really excited in 2023? Do you?
Me?
Yeah, we'll start with you.
Yeah. The most exciting news for the upcoming this year is, definitely, so there would be many novel biotech will show up in Alzheimer's cases, because finally we can see some of the real drug in Alzheimer. But I know some of the company already, although they're on stealth mode, but they do have some of the platform to overcome the recent antibody for the Alzheimer, the adverse event. So I can see some of the very interesting technology. Some of the small, but at least the reason why they cannot show or they cannot make a company, because there is no drug approved yet. So that's why they hesitated to start. But after some of the good news is recently that I can see the very clear movement in that field. So we can see the more exciting news in this year for that field. Not only for the big pharma like Roche or something, but some of small biotech can show the very, very interesting data this year.
Excellent.
Yeah.
Min, what are you looking forward to on the horizon?
So, for year 2023, it's going to be a full year, hopefully without COVID. We're back to normal. So, very excited to see everybody now, offline. And as I guess Michael have mentioned too, this is going to be a year where, investors and biotechs will find the right valuation. We will find the right opportunities to invest, and this is something that kind of interests me. But the key point is that when there is everybody knows the market will be tough still, but at least we know that it's going to be tough so that people can be prepared. So just don't be afraid of the bearish market. You guys are well prepared. It's worst thing they can do is you expect good things, but the market ends up to be very bad. That's the worst thing. So you're prepared. So, but just very excited to see normal life with the right biotechs. So those are some of the things that interest me in 2023.
Excellent, Michael.
Yes, definitely excited to meet people more and travel. We have a deployment pressure. We have to deploy $2 billion this year across equity and royalty credit, whatnot. So that's exciting part because we have to meet a lot of companies and we're excited to deploy. And we do have a mandate. We will do another investment in South Korea, so we're looking forward to that. We did a big deal in Hugel. I lived in Korea for 4 months last year for this. Thankfully, Korean government doesn't have to tax me. It was only 4 months. But so we're looking to do something in Korea again. We're opening our office in Seoul next month at Center One. So we're looking forward to meeting more Korean companies.
Excellent. Well, thank you very much, gentlemen. Do we have time for any questions, or do we need to?
We're actually on delay, but have some questions on the floor. That'll be fun.
Excellent. Any questions?
Yeah, everyone is thinking about the time.
I know. Yep, they are. Okay. Well, thank you very much, and gentlemen.
Thank you.
And Michael, now we will move on to the exciting first batch, the 6 companies pitching session, which will be hosted by Tom Dooley, partner from Sidley Austin.
Thank you, everybody. My name is Tom Dooley. I'm a Sidley Austin partner based here in San Francisco. I handle licensing and partnering deals. And I've been given the task of introducing the companies we're going to hear from in this first segment, and also the task of keeping things on time. So I would ask for 6-minute presentations, and then if you're going on, I might give you a signal, and we'll switch to a Q&A session from our investor panel over here for about 4 minutes. So 10 minutes overall each. I think we should maybe ask the investors to introduce themselves, if you wouldn't mind, to go down the line. There's a microphone, I guess, there. Sorry, is that right? Yeah. There's nobody new? Yeah, okay.
I'm new.
Okay. Yeah, okay.
Good afternoon, everyone. Good afternoon, everyone. I'm new. My name is Dan Wang, the head of Johnson & Johnson Innovation in Asia Pac. Very happy to be in this panel. Thank you.
Hi, everyone. My name is Fong. I'm from LYFE Capital. We are a global life sciences fund managing about $2 billion. Our team of 30 people sits across Menlo Park in California, Shanghai in China, and I'm from the Singapore office, just opened last year. So our Singapore office is covering the rest of Asia, and we're covering Korea as well. Very nice to meet you.
Go again, Michael Kim from CBC Group. We manage $7 billion today across venture, private equity, and private credit and real estate. Great to meet you.
Yeah, Jung again. And then, I'm charging over life science investment in KTB, and I'm happy to be here.
Hi, my name is Min from KB Investment. Great to be here. KB, one of the largest private bank in South Korea. Thank you.
That's nice. Hi, I'm Ted Kim from Oran Global. We are a life science focused investment bank as well as a healthcare fund. We're based in New York, and different from the panels, we bring 2 different perspectives as a fund and also from the banking perspective. Thank you.
Thank you very much. So the first company is PDC Line Pharma, and the presenter will be Eric Aliwa, the CEO.
Okay. Good afternoon, everybody. So I'm going to present you the activity of PDC Line Pharma. We're a cancer vaccine company based in Europe, in France and Belgium, based on a unique technology, which is a plasmacytoid dendritic cells. In fact, we are a spin-off from the French Blood Bank, which is a huge organization in France that collects the blood from transfusions. But we have as well the specificity to have academic lab associated. And the scientific founder of the company, PDC Line Pharma, Joël Pluma, has identified in the blood of a leukemia patient, a very rare leukemia affecting a subset of dendritic cells, the plasmacytoid dendritic cells. And based on the blood of his patients, he developed a cell line, GMP, approved, that we are still using today as a vaccinal platform. He was the first one to characterize this disease, which is a very rare disease. So since then, the company grew. We're a team of 32 people. As you can see here, we raised close to 56 million euros in dilutive and non-dilutive money, which is one of the aspects you can have in Europe. Our PDC line technology has 3 key advantages. It's a cell line of dendritic cells. It's immortalized, so it's very easy to expand. In fact, we can cultivate in a bioreactor, single-use bioreactor. We're at 10 liters bioreactor now. The origin of the cells being a plasmacytoid dendritic cell provide very interesting biological property, and I will show you some clinical data later. It's a versatile platform. We can address any kind of solid or liquid tumor. We just have to adjust the antigens that we stick on through peptide representative of the antigen that we stick on the cell line. We are a clinical stage. We are ongoing a phase 1/2 clinical trial in lung, non-small cell lung cancer, and previously academic feasibility study has been done in melanoma. This is the pipeline. Today we are focusing on the lung with what we call shared antigens, and we have at preclinical stage, as you can see here, the neoantigens, personalized neoantigens, and we expect to launch clinical trial over 2 years periods. The melanoma is not active anymore. Important to mention, we signed already a first licensing deal in Asia with LG Chem. I'm sure for some of you, you know, it's a large organization, but in life science, they are growing. And we provided the right in Asia for the PDC lung vaccine for the non-small cell lung cancer. And you see here the characteristic of the deal, $123 million plus royalties. We have, based on this deal and the contacts we made, we get the opportunity to have access to several already South Korean investors, including KIP, Korea Investment Partner, but as well UTC, Shinhan Financial Group, Alpha Holding, Hansong, and Brain Asset Management that are all members of shareholders of the company. We have a mix of Belgium and South Korean investors in the company. The team is definitely one of the assets. We have people with long experience, some of them coming from GSK, which is the case of our medical director, that has run the major cancer vaccine clinical trials of GSK. Claude Dedry, the CMC guy, has been involved as well in CMC, in cell therapy, including in GSK. Laurent Lévy, the CFO, has a long experience in oncology. And Joël Pluma, the scientific founder, joined full-time the company. And myself, I've already been an entrepreneur in several companies involved in IPO, M&A, in different organizations. So quickly, the clinical trials, the lung clinical trials, we are today have 4 cohorts of patients in non-small cell lung cancer. We are testing 2 doses, low dose and high dose of vaccine. First, alone, without checkpoint inhibitor. And the third and fourth cohort is with checkpoint inhibitor in associations, which is the pembrolizumab. So we have 6 patients for the low dose and 42 patients for the high dose. We have completed the 3 first cohorts, including the last one, the third one, which is the low dose with checkpoint inhibitor. And I'm going to show you the data that have been presented to ESMO, which is a big scientific conference in Paris a few months ago. As you can see here on the 6 patients where we have tumor and we can measure squeezing, shrinkage of the tumor, we had 4 patients with what we call partial response. Partial response is when you have more than 30% of the tumor that have shrinked. And for some of the patients, as what you see in green, it's close to 90%. So it's not borderline. Partial response is very strong response we get. And when you look the durability of the effect, meaning the shrinkage and how long the shrinkage stay over the time, it's up to 1 year period for this cutoff. The cutoff was in end of May this year. As you can see, the trend is extremely nice. We had 1 stable disease and 1 patient that did develop, as you can see here. So it's quite nice data. It's the first data for the low dose, and we're waiting for the high dose. The 42 patients, we are still enrolling and expecting to get the results. In terms of timeline, we should have the final clinical result of the current clinical trials. We are including the last cohort in May 2024, but we expect to have already interim data, which is a first step of the last cohort in August as well. And at this time, we could launch in 2024 new clinical trials that will be randomized clinical trials. And of course, raise new funds and develop further the company, maybe with new partnerships. I would like to thank you for your attention, and I'm ready for the questions.
Thank you for your presentation. It's happy to see you are addressing the most challenging cancer as lung cancer. Is there any rationale behind you chose lung cancer as the indication target?
Of course. In the beginning, when we had to decide which clinical trial to launch. We had an exercise, strategic exercise to identify and to screen different cancer. The criteria was first at this time, because we started the clinical trial in 2019, when we started to file the different documents. We wanted to have checkpoint inhibitor as a standard of care in first line. And secondly, we wanted a pool of patients where the enrollment will not span for 10 years, and to have a sizable clinical trial where today we're expecting 64 patients, and where we would have interest from key opinion leaders. So based on all these different criteria, we decided to go on for non-small cell lung cancer. And the size of this market is quite big, as you know. And the unmet need as well. The unmet need with the checkpoint inhibitor is still manageable. The difference, for example, for melanoma, where today with the combo, you have the bar is extremely high in terms of response.
So how long does it take to manufacture your product?
Okay, the manufacturing, it's quite quick. In 10 days, we have because we have a master cell bank, so we can take a tube of the master cell bank, seed single-use bioreactor. So in 10 days, you have billion and billion of cells. Then at the harvest, we split the cells according to the number of peptide representative of antigen want to stick. And after we irradiate the cells. But that is done in 1 day. What is longer is the QC, the quality control. And the quality control in total, we need 4 to 6 weeks to have the batches. But it's one batch for several patients. It's allogenic cell therapy, what we have here. And the objective for the next clinical trial is to go to 50 liters bioreactor, which is a quite big one.
Any sense of how long the cells stay in the patient?
Yes, good question. They are irradiated. So after 2 days, everything is dead. But, because the irradiations make the cells entering in apoptosis, but they still keep the capacity to present the antigens. So that has been demonstrated and shown in different models. But we know that we did study in animals, but after 2 days, all the cells have been destroyed. They are dead and nothing. But we infuse 6 times. So we infuse the cells weekly 6 times, and it's enough to go to the lymph node and to stimulate the CD8 T cells, to amplify what we can say to prime and activate CD8 T cells.
Okay, thank you very much, Eric. And thank you for staying on time.
Yes, please. I try to. Thank you.
So the next company presenting is AbTis, and we have Sang-Jian Chung, the CEO.
Thank you. Good afternoon, everyone. This is Sang-Jian Chung, the CEO of AbTis. Where is my slide? Okay, AbTis was founded in year 2016 and focuses to develop ADC therapeutics. And antibody drug conjugate called ADC is a targeted anti-cancer, consists of antibody, payload, and linker. AbTis has developed a linker platform and pipelines of ADC. Our platform linker technology, AbClick, is a site-selective ADC linker working for the most off-the-shelf therapeutic antibodies. So Claudin 18.2 targeting ADC called 8211 is a leading pipeline prepared by AbClick technology. So core competence of AbClick lies with the ability to site-selective conjugation to unmodified antibody with a defined DAR, resulting in a metabolically stable isopeptide bond to lysine 248 only. So our linker technology can adjust the PK property of ADC product. Also, this linker technology can employ diversity of payload, giving DAR of 2, 4, 6, and 8. And thus prepared ADC is very stable in serum due to the isopeptide bond. Then the scheme in the low side explain how our linker works. By simple mixing of our linker with antibody, it click functional groups transport the lysine 248, forming isopeptide bond. And then this click functional group further react with payload equipped with another click partner functional group to give DAR2 ADC. Then our Claudin 18.2 is an emerging target for gastric cancers because this Claudin 18.2 is overexpressed about 87% of gastric cancers. Then currently, about 20% of gastric cancers are overexpressing HER2, so Herceptin-based therapeutics can be used in this case. So we prepared ADC with DAR2 using anti-Claudin 18.2 antibodies, and then evaluated in vitro and in vivo. Surprisingly, our 8211 showed very strong anti-cancer activity against only Claudin 18.2 overexpressing pancreatic and gastric cancer cell line, and the potency was almost equal to the native drugs, MMAE. Whereas MMAE showed the cytotoxicity of cell lines overexpressing Claudin 18.2 or with cell lines without Claudin 18.2. Our competing ADC, zolbetuximab MMAE conjugate, showed 20 times lower anti-cancer activity against antigen-positive cancer cells. In vivo study showed MED of 8211 is less than 2 mpk. This is the almost the highest value. As you can see here, a single injection of 2 mpk of 8211 results in complete regression of tumor tissues in SNU 601 gastric CDX model. And then this table shows the comparison of key efficacy or toxicity of 8211. So MED was determined to be less than 2 mpk, and HNSTD here in the non-GLP monkey tox study resulted in HNSTD of 10 mpk. So the therapeutic index is estimated to be over 20. This is the highest therapeutic index among Claudin 18.2 targeting ADCs. For example, LM302, which licensed out from Lanova to Turning Point last year, they showed MED value of 10 mpk, but HNSTD has been reported yet. Also, our linker technology, AbClick, has been applied for radio immunotherapeutic agents, which can be used for companion diagnostics and also anti-cancer therapies. So we prepared radionuclide conjugated antibody using AbClick Pro and AbClick Standard, respectively. As you can see in the image, AbClick Pro product shows very long biological half-life, whereas AbClick Standard product showed very short biological half-life. This is very important for RIT. So the red line represents the radioactivity in blood. So AbClick Pro product shows very long existence of radioactivity, whereas AbClick Standard product showed this quick disappearance of radioactivity. So their biological half-life was determined to be 21 hours and 7 hours. So currently, this AbClick Standard product are evaluating for anti-cancer activity. Almost done?
Okay.
Yeah, so.
I just want to make sure you get time to answer questions.
Okay, yeah. So our technology has been applied for ADC, ARC, and other, drug purpose. And then currently this technology will be installed in ADC toolbox in Lonza, so anyone can use this technology through Lonza CDMO service. So, okay, so thank you for listening.
Thank you very much for the very interesting presentation. Appreciate that. And we also in our group, we're very excited on ADC and the technology, and we've seen a lot of activities and deals happen in this space, in addition to the linker technology. Peptide-based linker technology, there are quite of other more advanced stage pipelines already. How would you differentiate your linker technology compared to others, in addition to their other methodologies, such as a glycan, using a different way of using linker? So we'd love to hear about the differentiating factor.
Yeah. Okay. First of all, our linker can be selectively introduce drug onto the lysine 248 of a native antibody, not engineered or not modified antibody. So it can be applied for almost any type of therapeutic antibodies. So that's the, and then it includes only 2 steps of process. The reaction time is less than 12 hours. So we can prepare ADC in a week when we receive the antibody from the collaborators.
Sorry, I do have a follow-up question. Apologies. You've shown in a very impressive in vivo data. In the left lower panel, show that non-GLP tox study is coming in November or late. Any update on that? We believe the toxicity is a very important factor into an ADC.
Yeah, actually it shows a little gastric tox, but doctors say it's manageable. Then currently we are preparing non-GMP and GMP product for non-clinical and clinical studies. So it will be done this year and the next year for clinical trials.
Just a quick question on freedom to operate in your linker peptide technology in the lysine 268, and also on your financing history. I didn't hear about your investors.
We have gotten Series B funding until now, and then currently we are doing Series B bridge that we obtained around 40 million. But we are continuing.
How about the freedom to operate on your linker technology?
Right now was 40 million.
No, the IP, intellectual property, the freedom to operate in your...
Yeah, we have applied for patent in 8 countries.
Nothing issued yet?
No, not yet.
If there's nothing further, thank you very much.
Okay, thank you.
Okay, our next company is ElemediX, and the presenter is John Paul Rogers, the CEO.
Good afternoon. No. Okay. Good afternoon, ladies and gentlemen. Thank you for giving me the opportunity and for your patience to listen to me. I'm a bit of a strange difference in what has already been presented, because I'm going to present a medical device. So we started out of a frustration. I'm a medical doctor, and I sat together with an engineer, and we wanted to see if we can use physical treatment modalities to treat patients with advanced cancer. So we set up a company to use heat, so hypothermia treatment, to treat patients with advanced cancer, and very specifically our first focus is on patients with pancreatic cancer. So pancreatic cancer is, and to anticipate the question, why pancreatic cancer? Pancreatic cancer is a very, very difficult disease to treat. It's a very significant unmet medical need. There is not so much changed over the last 40 years in the survival rate of patients with pancreatic cancer. And while more personally, I lost my mother to pancreatic cancer 30 years ago, and if I compared what's happening today and what's happening when she was ill, there's actually not so much difference, except for a very small population of the patients with BRCA1 or 2 mutations. We also know that most pancreatic cancer patients will develop chemoresistance, and also that immunotherapy is not doing that much at the moment. We also know from previous clinical trials that pancreatic cancer is sensitive to heat, and that heat can change chemoresistance in these patients. And that's why we developed HALT. HALT is the highly accurate long-term systemic thermotherapy treatment platform, where we have developed a thermal heating, an incubator based on patented thermal heating method. We have developed a patented thermal model. We have developed a patented sensor, and we have developed a patented predictive biomarker set to predict heat sensitivity in the patients. At the moment, we are in first-in-human trial, so we went through all the preclinical data, and well, I didn't take them with me, because usually most investors are not really that interested, but they're all published. So and of course I can show them. We are at the moment we also went through all the safety studies with animals, and now we are in first-in-human studies in the University Hospital in Antwerp, where we have already treated 5 patients. It's mainly a safety study, of course. We didn't show any significant side effects of the treatment, and these are all-comer patients, so they're very difficult to compare results. But we have already shown that in 2 of the patients there was a very significant drop in the tumor markers. We are setting up 3 new sites, or 2 new sites actually, in Europe at the Amsterdam University Hospital, in the Netherlands, more specifically to speed up the recruitment, and very specifically also in Singapore General Hospital as a second site to start treating Asian patients, and also to start the safety issue and safety data on Asian patients. We target to hit the market in 2025. We are in first-in-human at the moment. After that, we foresee a pivotal trial with 30 patients. And the main reason for me to come out to San Francisco is to look for funding for the pivotal trial. And then of course, we need a larger trial to get market approval. Perhaps you know, but the total addressable market of pancreatic cancer is very significant, approximately 500,000 new patients yearly. But the technology is not limited to pancreatic cancer. And while we have tested already on colorectal cancer and melanoma, and it can work to these as well. There is a lot of data, for instance, on cervical cancer. So it's not limited to one specific tumor type. And there are also non-oncological indications like major depression, which also react very well to heat treatment. The business model is based on the recurrent sale of a disposable sensor kit with a very high margin. The company is led by an experienced management team. We have 18 people on the payroll at the moment, and we have a management team including all the most important roles, and with people who have a lot of experience. Financing, well, we have already received 10 million in seed money over the last 3 years, plus 1.7 million in non-dilutive grants. We are open at the moment for a Series A of 7 million, more specifically to fund the instrument development and the initial pivotal trial. And then, of course, we need an additional round of, we estimate 25 million to pay the extended pivotal trial and to go through the regulatory process. We have also been named in the top 5 pharma startups in the pancreatic cancer space. I think I even saved you a bit of time.
Thanks, interesting talk. It's good that you're doing simple clinical trials. I think it's one of the places that can really get Asian patients. But my question is mostly actually about the technology, because most of the time you see that the hypothermal treatment is always very local. Yes. But this is the first time I'm seeing a systemic hypotherapy. Can you talk about the heating process and how you monitor for safety in this case?
Yeah, so you are right. So most of the currently available, commercially available hypothermia devices, which are heavily used actually in South Korea, is local hypothermia. And that's usually combined with radiotherapy. So they give hypothermia and then they give radiotherapy, and the hypothermia increases the sensitivity of the tumor for radiotherapy. My dream was, I'm a medical doctor, so my dream was to treat patients with systemic disease, with metastasized disease. So when we started our quest and saying, what are we going to do? I said, it has to be something which we can apply systemically. So immediately we went to systemic treatment. There are at the moment 3 companies in the world who are doing very similarly, well, to others and us, and they are using other technology. We have also tried that. It's not safe. It's an American company and a Dutch company. We know that it's safe. We've seen that in the animals, but we've also seen it in 5 patients. The patients are under full anesthesia during the treatment, so they don't really notice anything. And the only thing they report after the treatment is fatigue. So there is fatigue for, let's say, 2 or 3 days, which is very comparable by running a marathon. So, well, I can't run a marathon. I have no idea how it feels, but I think it gives you a lot of fatigue. But it is like heavy exercise fatigue. So that's the only major complaint we hear at the moment.
Sorry, so what's the heating technology used?
Well, we actually use hot storm. So we use it's a hot air which is flowing in a very high velocity over the patient. The main difference, what we do, and we do different to the others, and which is actually also in one of our patents, we have 6 patents, 2 of them are granted worldwide. The most important thing is that we can not only heat, but we can also cool. Because if you heat the body up to 40.5 degrees, you get a catabolic reaction, and the body is actually starting to produce more heat. And then actually you have to cool the body. If you don't do that, then the patient burns himself. So that's something, and that wasn't done in the past. And that is probably the explanation for a lot of accidents which happened in the past.
Thanks.
One interesting side note, and because I still have a few minutes, is that we are also looking for partners, because we also know that if you apply heat to a tumor, the tumor starts to react differently to immuno-oncology, starts to react differently to chemotherapy. So, for instance, BRCA1 and 2 non-mutated tumors start to behave like BRCA1 and 2 mutated tumors if you heat them first. So that's also interesting. It could be interesting for biotech companies that don't see the number of results they want to see. Perhaps we can try together to make their results better and push my device in the meantime. Thank you.
I have a question for you, actually. Since you saved me a minute, I'll take it. What are your thoughts about the economics, the costs of a medical device therapy for something like pancreatic cancer as compared to therapeutics?
Well, the cost of the device is the device is not the problem. It's the business model is based on the recurrent sale of the kit. And of course, the price of the kit we can put wherever it's needed. At the moment, I benchmarked it, and it can be something around 5,000 euros if you compare it with radiofrequency ablation needles, for instance. And if you have to apply it 3 times, then you have a price tag of, including the OR, let's say 50,000 euros per patient in total cost, which is very similar to the price of an operation. Surgery is approximately the same price.
Okay. Thank you very much.
Next company presenting is FORTUGA Bio, Sungjun Yoon, the CEO.
Hello, Sungjun from FORTUGA Bio, and I thank everyone here for this wonderful opportunity to present. At FORTUGA Bio, we're developing an off-the-shelf type dendritic cell-based cancer vaccine. And before we go into the how, I want to first emphasize why this is important. Today, we're going to talk about cell therapy, or more specifically about dendritic cell therapy. And dendritic cell has a great potential in treating cancer since they work at the very upstream of our body's entire immune system. But at the same time, they pose 3 major problems when it comes to commercializing them as commercial drugs. The first is the complexity of the treatment process. Since the number of these cells innately present in a patient's body is so small, they have to be extracted and injected back into the patient's body for every treatment. And since we're dealing with living cells, they can only survive and stay active in the patient's body once they're injected for up to 48 hours. So our approach is to first develop a cell line that's immortalized and also allogeneic that can be mass produced. And the second is to synthesize nanoparticles using the components of this DC cell line, which is much more stable in comparison to the living dendritic cell or the conventional DC therapy. The second problem is because of this complexity in the treatment process, the manufacturing cost is very high. It costs around $1 million per patient per year. Most of them arising from the material and labor cost. By mass producing these cells and synthesizing the nanoparticles, our goal is to reduce this manufacturing cost by at least 80% of the original. And the last problem is the coverage, which is not only limited to dendritic cell therapy, but immunotherapy in general can only cover up to about 30% of the entire cancer patient population. So what we're proposing is that our platform technology can easily allows fast and easy combination of different types of immunotherapeutic agents that are already commercialized into a single compound, in which case we can target different indications that were previously very difficult to treat. So this is the overall scheme of our approach. We are first developing an immortalized cell line that can be mass produced, and these cell lines will, using the membrane of these cell lines, we synthesize nanoparticles which can be stored for a long period of time. And just before injection into the patient, these nanoparticles can be tailored and personalized for the patient's need. To go into a little bit more detail, we call these nanoparticles artificial nanodendritic cell, and the reason why is because these nanoparticles can mimic the main function of a living dendritic cell, which is antigen presentation. And on top of this antigen presentation property, we can also conjugate immune checkpoint inhibitors such as CTLA-4 or anti-PD-1, which also provides an additional T-cell activating property. I'm emphasizing the stability of this material a lot. We also have tested the stability in terms of their maintaining their efficacy. We stored the nanoparticles that were synthesized at room temperature for 30 days and injected them back into the T cells, and we saw that the level of T cell activation has been maintained. It's same as a freshly synthesized nanoparticle. The most important part will be the tumor regression. The first row here, you see we're comparing the nanoparticle directly with the conventional DC therapy. And we saw that even without the immune checkpoint inhibitor conjugated, we still see the same level of tumor regression as living dendritic cell. The second part is if you conjugate these nanoparticles with anti-CTLA-4 antibody, the tumor regression level is further enhanced. And in fact, about a third of the experimental group, we saw a complete removal of the tumors. The last row is another tumor model, which is more aggressive and therapy resistive tumor model. We see a similar trend of tumor regression as the previous result. So just to point out some of the major milestones that we have for the next few years, we are currently developing the cell line that I proposed. We will be finishing them about by the end of this quarter. And since this is a very complex material, even from the beginning, we're focusing a lot of our attention on the CMC and optimizing the quality of the nanoparticles. And once it's done, we are planning to submit for IND by end of 2024. And as for the financials, we just completed our seed funding last year, and currently we're raising our pre-Series A funding, which we plan to close by end of March. Lastly, I just want to give you an overview of our company. It's a very newly established company. It was founded in September 2021, so just over about a year ago. We're working with 2 university professors from Yonsei University. Professor Ha is specialized in immunology. He's responsible for studying the mechanism of the drug. Professor Hong, he's part of the chemical engineering department, so he's responsible for optimizing the mechanical properties of the nano material. And we also just licensed in 10 patents covering the methods and the usage of the nanoparticles from Yonsei University. That will be all for the presentation, and if you have any questions, I'll be happy to answer them. Thank you.
Very interesting talk. Appreciate being here. So quick question. I mean, I like the idea what you mentioned in terms of the economics factoring in terms of how dendritic cell could be commercially viable, knowing about Dendrion and other experiences have been. And you mentioned about the cost. How much does the gold nanoparticle, as well as combining with checkpoint inhibitors, add to the cost gain?
Actually, that one I haven't mentioned. The gold nanoparticle is right now is being used as sort of a control substance, just providing the mechanical support for the membrane. But right now we're planning to change that to zinc oxide, which is already approved by the FDA. And also for the next step, we are trying to utilize a biodegradable polymer, in which case we can embed additional type of drugs like mRNA or cytokinin, in which case the nanoparticle would act much more like a living cell than just being a synthetic material. As for the cost, we haven't really figured out about the solid nanoparticle, but for the antibodies, I guess there will be two different strategies. We could work with the companies that are already selling these antibodies, or another strategy will be once we have the stable cell line, we can express these types of antibodies on the surface from the cell level, and we can create and synthesize the nanoparticles using those cell lines. In that case, we'll be able to save the cost for the antibodies present on the surface.
And do you think this platform, what you've created, is it only applicable to dendritic cells, or can it be used for other cell therapies?
Right now, I think it's mostly applicable because dendritic cells they don't have a direct killing mechanism. Their main function is to present these antigens to the T cells or other types of immune cells. But it's not limited to dendritic cells. It could be applied to other types of cells as well. And also another thing is we don't even have to use the immune cells in this case. We could use just completely different type of cell which express the peptide or the ICI antibodies, and we can still make them as nanoparticles. But we haven't tested that method yet, but that could be an alternative to our main approach.
Thank you.
I'm really interested in that coating technology you use to actually make, is it really a phospholipid bilayer around the gold particle?
Yes, yes.
With proteins embedded?
We actually tested if it's the right side up. I mean, if the surface molecules are actually present on the outside. So, yes.
And in terms of quality, how do you do QC on this kind of product?
Right now, we're using mainly two different methods. First, we have to visualize them using TEM because the size is very small. It's about 100 nanometers in diameter. And to quantify these particles, we use zeta potential and DLS to sort of get a graph to get the distribution of the size of the particles.
I guess that would be good enough for clinical applications?
Yes, for like exosomes or lipid nanoparticles that were previously used, we're trying to follow the same guideline.
Yeah, very quick question. Are you open for collaboration at this stage? If yes, what type of partner are you looking for?
We are always open for collaboration, except frankly speaking, I don't think the technology is mature enough where we can license it out to another company, but in terms of collaborative R&D project, we're always open. One thing would be, I'll say two things. First is if we can work with a neoantigen discovery platform company, and we can embed those information on the particle to find a new target. And the second will be, like Michael asked before, the antibody. So if we have a company that produces these antibodies and they're new, like other than CTLA-4 or PD-1, that could be another approach that we can take. Yes. Thank you.
Thank you very much. I see there are more questions, but perhaps after the presentation.
Yes.
Thank you.
Thank you.
Thank you very much. I've had the privilege to work with the next company you're going to hear from, Imagin Ab, and the presenter is Ian Wilson, the CEO. Ian?
Thank you very much, Tom. It's nice to meet you in person. Yeah, so we actually did a deal by video, so that was very good. Hi, I'm Ian Wilson. I'm the CEO of Imagin Ab. We're a biotech company based in LA. We have no products approved, but we actually are revenue generating, which sounds bizarre. So we work in the field of mini-body technology, so fragmented antibodies, and we develop imaging agents, and we develop radiopharmaceutical therapy. So the previous talks have talked about the importance of biotherapy, and we work in that space from an imaging point of view, and we're developing next generation radiotherapy. So currently we have $26 million in revenue from pharma companies who are using our technology, and we use that along with money we get from kind investors to progress our pipeline. So it actually feeds that. Our platform technology, which we own all IP rights to across most geographies, including Korea, allows us to humanize mini-bodies, which are stable and immunologically inert. Our imaging agent is the first in class. It is the only imaging agent that you can use around the world. We supply it in North America, Europe, and Australia, and we will be able to supply it in Korea by the end of this year for pharmaceutical companies to use it in their clinical trials. We've took that technology and we've started an early pipeline. We're just completing preclinical studies, and we'll start IND-enabling studies this year to enter the clinic in radiopharmaceutical therapy. And we've got a good supply chain to allow us this to happen. We're currently funded by AdAge Capital, Merck Sharp Dome, the Parker Institute, Raptor, Innoviva, and Cycad Group, and they've been very loyal investors. Their investment has been purely, in the first instance, on our CD8 agent, which I'll talk about first. And we're now looking to raise around $50 million to get us through phase 1 and phase studies for our radiopharmaceutical therapy pipeline. So I can just skip on this slide. So what do we do? If you want to design an imaging agent, it has 3 components, and the same is for a therapy agent. We have binders. You add a chelator that can bind a radioactive metal, and to that you either add an imaging radiometal, in our case, zirconium-89 or copper, or you use lutetium-177 for therapy. And you generate a targeted specific agent that targets those cells either for imaging or actually to kill the tumors. We've been doing this for many years, for 15 years, so we've got a strong team and we've took many through clinical trial. So first of all, I'll just skip through our imaging agent that images CD8 T cells, and I think some of the presentations showed you the value of that in the IO space. We've generated a mini-body. It's called crefmilimab. It's radio-labeled with an isotope known as zirconium-89, which means it actually has a half-life of 3 days, and the product has a life of 6 days. So anyone who works in imaging, that's quite a long time. So it allows us to have a very robust supply chain. So we can manufacture it in one center in the US and distribute to all North America to any clinical site. So again, that's what we will do in Korea. We'll just have one manufacturing site and cover the country, unlike 18F agents where you have to have a complex supply chain. And what does it do? Well, this is what it does. You image with a standard PET camera. There are thousands in the world. Major hospitals have them. So we're now in phase 2B, and this is an image of a patient who actually has got melanoma. On the right-hand side, you can see an image taken with radioactive glucose, and you can see that the glucose has bound the tumor. Actually, on the left-hand side is an image highlighting every T cell in that patient. So you can see the T cells in their bone marrow, in their long arms, in their arms and legs. You can see it in the spleen, and you can actually, if you look at the neck, you can see the lymph node. And in this case, you can actually see some T cells in the leg, just next to the tumor. Actually, the lines going through it, so you might miss that. So actually, what you can do is, why do people want to do this? Well, biopsy is very difficult. Metastatic patients can't shove a needle all the time into a patient. And actually, you can only do one tumor. You can't do it all the time. Replacing that with imaging, you get a head-to-toe distribution of all T cells with one image, and you can bring them back. And what does this mean in real terms? So in real terms, you can see in this image here, you can see the baseline image. You can, what I showed on the first image, if you go down to the bottom, you can see the T cells are just distributed around the edge of the tumor, which means the T cells haven't entered the tumor. After one cycle of of checkpoint inhibitor therapy, you can see with the bottom panel, the scan looks more intense and it's more uniform. So showing that the checkpoint inhibitor has done its job. And we see that in 1 month compared to CT, where you're looking at tumor shrinkage. In this case, it took up to 4 months to see that. So if you're a clinician or a pharma company and you want to know if your drug candidate working, we can help you do that a lot quicker and actually prove your mechanism of action. And we can do that with cell therapy. We can do that with normal drugs. Sidley actually helped us license this technology to Dongcheng, the second largest pharmaceutical company in China. We did that but virtually, so during COVID we actually never met, and we never met Dongcheng for 2 years while we did that deal, so that was quite encouraging. So we've done that, so we've been able to do that. And we've progressed our clinical trials and we'll be on market in 2025. But we've got over 20 companies using our technology, ranging from Boehringer Ingelheim to the next generation companies such as BriaCell and Parthenon, all wanting to use our technology to help them with decision making. So that's CD8, and we get revenue from that, and it's pretty spectacular revenue. Our profit margins are between 75 and 80% profit margin on those deals. We expect to increase those revenues, and at FDA approval, the market size is around 600 million for the US. I've got very little time, but I'm going to talk about radiopharmaceutical therapy. We're very strong in this because of these 4 areas: supply chain, novel targets, and proprietary platform. Anyone can work in RPT, but what we've done is take a top-down approach. If you're going to develop something like radiotherapy, it's actually slightly complex because you've got to radio label it. It's got a bigger ask than a standard drug, so you've got to be special. So this table shows 3 points. All trials in radiotherapy today, trials and drugs approved in therapy, and actually the outcome of those patients. So what is the ideal situation? Well, the ideal situation is to work in a space where there's low competition and actually the survival rate is low. So if you look at this table, you can see cancers such as small cell lung cancer and ovarian cancer. There's little competition for drugs in that development, not many on the market. So that's our focus. Our focus is to develop next generation agents for small cell lung cancer and ovarian cancer. Where are we in that process? As I said, we've got intellectual property. We are currently entering IND enabling experiments this year, and our goal is to get that in the clinic in first quarter 2024. We developed a number of years ago a prostate imaging agent and also radiotherapy. It's a very competitive space. As a small company, we didn't think that was where we should be. And actually in December, we out-licensed the diagnostic version of that agent to a third party, which we at this time cannot disclose. So what are our funding milestones? We have 3 value inflection points, 1, 2, and 3. We've achieved 1. Our goal is with 50 million is to achieve value inflection point 2 and 3. We can tranche that investment. Our goal is to show a readout at the end of a phase 1, phase 2 experiment, late 25, 26. We've got cash in hand from the revenue we go. We want to raise 45 million to complete the RPT experiments and around 10 million to get the CD8 agent to market. On that, I will say stop. So thank you very much.
Thanks, Ian. Hi.
Very impressive. Very impressive to see the PET especially. Just two questions on that. I'm not sure if I've mentioned, how would you target the CD8 cells only, not the other ones? That's the first. Oh. Okay. And the second question is, I know you mentioned that you have a longer shelf life than FTG, which is impressive. But 6 days still short, and you will have to have. You currently have 3 manufacturing sites. How would you? Multiple manufacturing site is inevitable in the future. So how would you control the quality?
How do we control the quality? Yeah, so to achieve sales in the US on approval, we probably need 3 manufacturing sites to meet demand in the first 5 years. So not a lot. So what we've actually done is that we've invested in an automated platform. So the radiochemistry is automated. But before that, the precursor, which is a biologic, is actually uses the same quality standards as ADCs. And also the conjugation, actually we use contract houses who are used to doing ADC. So we use the same quality release criteria as someone who develops a therapeutic.
I think we're at about time. Sorry.
Go ahead. It's more of a capital allocation perspective. I've seen that your revenue generation of the first asset requires $10 million. With great margin, it seems like that business model should be expanded and capital should be more flowing that to really grow that. But significant chunk of the money raising will be going into the radiotherapy. From a capital allocation perspective and with the time to ramp, is there a reason behind the priorities of those?
Yes. So we only actually need 10 million to get it to market because of the revenue. So although we're revenue generating, we burn more money than we currently have. But in 2025, we will have more money. So in 2025, we become self-funding. So the funding is really to cover the next 2.5 years. And once it's approved or out-licensed, then it requires no more funding. So really, it is the last round that we perceive we need to move this forward. Does that answer the question?
Thank you, Ian. Okay, the final company in this segment is GI Innovation, and the presenter is Nari Yoon, who's the Executive Director and Head of Clinical Development. And I think your slides will appear over there. Yes. Okay.
I'm very sorry that we changed the slides at the last minute, so the slides will be shown there. So we wanted to share the updated clinical data with all of you today. So that's the reason why we changed the slides. Thank you for the operation team today. So you can see the slides over there as well and in front of here. So yeah, thank you for your understanding. So it's a great honor to present today. I'm Nari Yoon, Head of Clinical Development. Since founded in 2017, we've been one of the fast-growing biotech in APAC region. So far, we've raised $210 million and now in preparation of public listing. During the last 5 years, we've made great achievements and we are building our presence in the industry. So we are developing biologics for patients with cancer and allergic disease. So today I'll be focused on GI101 first, the most advanced program. So as we all know, checkpoint blockade, especially anti-PD-1 antibody, such as pembrolizumab, showed promising clinical results and thereby being a standard of care for around 30 types of solid tumors. Despite of the fact that average response rate is still low, approximately 15 to 45%, and therefore there is still huge unmet needs to improve the outcome. I'm so sorry, it's not really working well. So why PD-1 antibody does not work for some patients? PD-1 is an immune checkpoint protein expressed on the immune cells. Clearly, anti-PD-1 antibody does not work if there is no target that is PD-1, for example, in situations like lymphopenia. In fact, lymphopenia is known as critical factor for determining the response toward the anti-PD-1. This data from the Johns Hopkins, 167 patients received nivolumab or pembrolizumab. The data shows that if patient baseline lymphocyte count is below 1,000, their progression-free survival was shortened. In addition, if patient experiences post-treatment lymphopenia, prognosis was strikingly bad. This is really well demonstrated in many solid tumors, including lung cancer, melanoma, showing survival difference of lymphopenia versus normal patient on anti-PD-1 treatment. GI-101 was designed to overcome immunotherapy resistance by increasing lymphocyte. It is a novel immunocytokine combining CD80 to interleukin-2 variant, and by having CD80, GI-101 is able to target CTLA-4 expressing immune cell and tumor cell, thereby reducing the off-target effect. This leads to delivery of sufficient amount of interleukin-2 to the target, which ultimately leading to lymphocyte expansion around the tumor. So what happens if there is only long-acting interleukin-2 variant without CD80? As you can see here in the pink line, the monkey-injected FC interleukin-2 variant only significantly lost their weight after the injection. This is similar to the observation with the pegylated interleukin-2 in human. They were not able to dose sufficient amount of interleukin-2 due to the dose-limiting toxicities. However, GI-101 did not result in body weight loss in monkey, showing that CD80 is responsible for better tolerability. Indeed, in our clinical study, GI-101 was escalated up to 0.3 mg per kg, that is approximately 10-fold higher dose level compared to other interleukin-2 agents. The underlying mechanism of this is reduced off-target effect by targeting tumor and immune cells. Mice with a tumor received radio-labeled GI-101, and at 48 hours, we saw that GI-101 was mainly distributed to the tumor as well as secondary lymphoid organs like spleen and lymph node. It works finally. So in our ongoing clinical study, we are seeing that GI-101 as a single agent and in combination with the pembro increases lymphocyte expansion significantly up to 4-fold from baseline. From the historical data from the proleukin, responders show the high lymphocyte expansion, while non-responders show little or no lymphocyte expansion, indicating the importance of lymphocyte expansion in anti-cancer activity of interleukin-2. Now, is GI-101 showing clinical activity? The answer is yes. While the other competitor of interleukin-2 space did not show any objective response in first-in-human monotherapy setting, GI-101 monotherapy exerted one confirmed partial response and one unconfirmed partial response, both in immune checkpoint blockade refractory setting. One patient with the checkpoint blockade refractory cervical cancer even showed a 100% reduction of target lesion only with the GI-101 monotherapy. In addition to that, we also saw significant tumor reduction minus 30% in one microsatellite stable colorectal cancer patient with liver metastasis who is currently on treatment for more than 10 months. With monotherapy activity, we are seeing synergy by combining with the pembrolizumab. Among 16 patients, 2 partial responses were observed, and many patients are ongoing the treatment. We wanted to understand the lymphocyte expansion by GI101 is correlate with the clinical outcome. When we analyzed progression-free survival using AI, the group with the highest peak lymphocyte level showed the longest PFS. We did observe similar pattern with the peak CD8 T cell level, indicating the correlation of either lymphocyte expansion or CD8 T cell expansion and PFS. Finally, I would like to show you striking results from combination of allogenic NK cell and GI101. By combining GI101 to NK cell, it persisted up to 4 weeks, while Proleukin only persisted in NK cell less than a week. In conclusion, GI101 is an immunocytokine that can overcome lymphopenia and immune cell persistency and has a potential to be a game changer as an immunocytokine. So I would like to move to the GI301 for allergy program. GI301 is an IgE trap protein containing Fc epsilon receptor 1 alpha, the endogenous IgE receptor. Because GI301 contains endogenous receptor, it has high affinity for IgE that is 72-fold higher than that of only approved anti-IgE drug on the market, omalizumab, also known as Xolair. Even with the several pitfalls, omalizumab is a blockbuster with a sales record of $3.4 billion. However, due to the low binding affinity to IgE, omalizumab does not work in all patients at all, in patients whose IgE level is over 700 IU per ml, and also it does not work in the patient who has autoantibody. GI301 can overcome these limitations by having endogenous receptor. In an ongoing phase 1 trial operated by trusted partner Yuhan, GI301 exerted immediate and complete suppression of free IgE by single injection, while omalizumab did not, showing the clear differentiation compared to omalizumab. With this strong pipeline, we expect next few years will be exciting, jumping up moment for the GI innovation. For patient and their family, we will focus our capabilities to maximize clinical benefit of our asset, exploring different combinations with advanced treatment modalities. Thank you for your attention, and I'm happy to answer any questions you might have.
Great. Thank you for the big effort you made to present the latest data. It seems like you guys have a lot of pipeline. So quick question, I think it's very interesting about doing the bispecific. I think neutropenia, as you mentioned, it could be an important, lymphopenia could be an important part with the CD80. But obviously with the Nectar BMS spectacular failure, there's still 12 IL-12 guys out there. Do you think that's the right second antigen to have? Or do you have others in the pipeline that you may combine with the CD80?
So actually by having CD80, we have a totally different characteristics compared to just conventional IL-2. So just looking at the recommended phase 2 dose, we escalated up to 0.3 milligram per kg, which is approximately 10-fold higher than Nectar or other second-generation interleukin-2 agents. So that means the problem of IL-2 is really that we cannot deliver the sufficient amount of interleukin-2 to the tissue or even in the tumor. So in that regards, if we can deliver the sufficient amount of interleukin-2 to the tumor, that will be great agent in terms of the anti-tumor activity. So we are thinking that CD80 combined IL-2 has a strong potential to be even not really different from these just conventional with interleukin-2.
Thank you very much, and thank you to all the companies who presented in this segment, and to the investors for their interest and their questions. I'd like to hand it back to Jury right now to introduce the next segment.
Okay, thank you, Tom. I've been waiting weeks for this case study. It's a popular deal in Korea. It's called Janssen and Yuhan cross-border deal. All the presses have been writing on their headlines, and this is something that I've been waiting for. I will have Josh Hofheimer, co-head of Global Life Sciences Practice at Sidley, Austin. Dan Wang, head of J&J Innovation Asia Pacific, and Taejin Yoon from Yuhan Corporation. He's the executive director. He'll be up on the floor. Please.
Why don't you guys grab a mic?
Do you need one here?
Grab a mic. So, thank you everyone for joining us for this session. I know we're running a little bit long, so we'll see what we can do about making up a little bit of the time, but this isn't too long of a session anyway. Pleased to be here. As was said, my name is Josh Hofheimer, and I help lead our global life sciences practice, and actually started my practice, like Tom Dooley, is very much focused on collaborations and licensing. And among other things, we started our Korean life sciences initiative about 5 years ago now, and one of the sort of leading deals was actually representing Yuhan in the transaction that we're going to talk a little bit more about with Janssen. But first, before we do that, why don't I give each of my panelists just a minute to give a little background on themselves. We'll hit a few general questions about their partnering experience, and then we'll dig into the transaction itself. Go ahead, Don.
Hi, nice to meet you. My name is Taejin Yoon. I'm heading the Global Corporate Strategy Group in Yuhan Corporation. And my background is chemistry, biochemistry, and immunology. And now I take care of the global licensing in and out and all the collaboration and some investment. Nice to meet you.
Hi, good afternoon. My name is Dan Wang, the regional head of Johnson & Johnson Innovation in Asia Pac. Very much thanks the organizer to inviting us to be here, offering the opportunity to connect. Actually, I got to know Taejin 4 years ago, back to 2018 when we made the collaboration. So for myself, I'm leading the Asia Pacific Innovation Center located in the major market in Asia Pac, Shanghai, Seoul, Tokyo, and Singapore. So our team is looking for the most novel science and the partners to connect them with within the Janssen expertise, like the discovery, development, commercialization, regulatory, etc. So we are here for more than 8 years in Asia Pac. So in J&J, we believe the good health is the foundation for people's life. That's why we invest so much to pave out the way for innovation by collaboration. So in the next 30 minutes, I'm very happy to share some of the details for the Yuhan collaboration and also to share the unique model for Johnson & Johnson Innovation and our strategy.
Thank you. So why don't we start, Taejin, with just giving us a brief overview of Yuhan's open innovation strategy and your global innovation strategy.
Yeah. Before that, I just wanted to introduce the Yuhan Corporation because there are some of the Koreans, but some others. So Yuhan is the number pharma in South Korea in terms of their sales, and founded in 1926, so almost 100 years old. And we looking, even though we are a local top-tier pharma, but we wanted to be a global pharma in the near future. So in that purpose, in R&D side, so from the early discovery to at the end of the global approval and go to the marketing. So global strategy of the global open innovation, what we are focusing on is compensation of what compensation of the weakest point what currently we have. So we relatively have low experience in the global clinical trial and go to the approval and go to the market. So main outbounding strategy is we looking for a partner who can who have extensive experience in that range, then we can collaborate further. And in other side, we also very interested in some early collaboration because we are not academia, so we still have some deficiency in terms of the cutting edge technology and some MOA study and some basic science. So in that point of view, we also looking for several global university and college and research institute. We can collaborate with build up the potentially first-in-class and best-in-class pipeline. So at this moment, we still working on some research collaboration with Imperial College in London and some platform technology collaboration with PIs in University of Cincinnati. Yeah.
Yeah, let me just stick with you for a moment, Taijin, because what you just described is, impressive both in the commitment, by Yuhan to develop and cultivate, internally and through in-licensing arrangements at the earliest stages, university academic labs, and many early stage companies in this room as well. And then converting that to outbound licenses when appropriate and when ready, and outbound collaborations. And I mean, look, we've had the good fortune of, I think, working on 3 or 4 with you all, including Boehringer Ingelheim and Gilead. Tell me a little bit about what's the value proposition for you that's the big driver in who you select for these partnerships, because you've had different partners with each of your different therapeutics.
Well, in terms of the outbounding strategy, we're looking for a company who has a lot of experience in specific indication and a wide network with KOL in that specific indication and therapeutic areas. That will be compensate for our weakest point. So in other words, inbounding strategy, if collaboration with the academia is very early model, but regarding the biotech company, we were focusing on some asset is almost ready to the IND or 1 year before the IND, then we can speed up the data package for the IND, then move on to the clinical stage. That is a good timing. But sometimes, depending on the therapeutic areas like CNS and some other things, we also prefer some early collaboration because it really necessary to have a platform technology and some novel approach is needed. So in that case, we doesn't have a room to compete with other pharma and global pharma until they have a full package for the IND. So that is the timing what we're looking for usually.
Thank you. Don, maybe you can comment a little bit from the similar question from your perspective on what J&J Innovation is looking for in its decisions to partner, and especially with the APAC region where you cover.
Yeah, so before I answer this question, just want to provide a little bit more details about Johnson & Johnson Innovation. We believe a great idea can come from anywhere in the world. So depending on where the ideal concept is in the life cycle, we leverage our full spectrum of capabilities to engage the partner. That include external collaboration, early partnering through the innovation centers, and also the startup incubation by G-Labs, and also the equity investment by JJDC. And if the asset is in the late stage, we have the business development team to engage with the partner. So to leverage this full spectrum of capability, we actually work with the partners at different stages. No matter it's an early stage, a preclinical lead compound, advanced lead, or maybe phase 1 or phase 2 candidates, as long as the asset is within the strategic focus for Janssen pipeline, and there is a synergistic capability that we can pursue, we can work together, we will be very happy to engage with the discussion. So in terms of what we are looking for, I would say pipeline asset in the major therapeutic area of Janssen pipeline, such as oncology, immunology, neuroscience, infection disease, cardiovascular, metabolic disease, etc. And we are looking for the novel asset, but also we have some existing program that we are looking for the additional component that can help us optimize to deliver the best molecule to the patients. So that is also what we are looking for, maybe some platform with a novel technology that can help us to address our medical needs.
And that's actually a great lead-in. Thank you very much to a little bit of detail about the Yuhan-Janssen relationship and the deal you all did, because in some ways it was looking at a component to help you all build your own therapeutic. This was a deal for people, just briefly, it was struck in late 2018, and it was a collaboration and out-license of Yuhan's proprietary therapeutic lazertinib that was initially targeted for treatment in non-small cell lung cancer. But it is a robust compound, and the idea behind the collaboration was to explore both monotherapy applications and combination therapy applications with Janssen products. It was a $50 million upfront, which was a nice deal for Yuhan. There were more than $1 billion in potential milestones, both development and sales milestones, and tiered royalties that tiered up to low double-digit percentages. Or sorry, low, just a little more than 20%, so percentages. So with that, tell me, and you also have the good fortune of having two people that were part of the original scientific teams on both sides at the time. Tell me what brought you all together. What was this opportunity that you saw in, Dan, that you saw in Yuhan, and Taejin, that you saw in Janssen?
So when we licensed in this molecule from the Oscotec, it was not a second-tier compound. There was a highly competitive landscape regarding the third generation of EGFR TKI. So we have to find the right strategy, how we can fastly develop this as soon as possible and move on to the clinical stage. And also, at the same time, what could be the best partner who can develop globally and go to the market. So in 2016, if I remember correctly, 2016 AACL, our scientists saw the presentation of Janssen team. They do the combo therapy of the Abion plus Tagrisso. There was a synergistic effect, and after saw those results, our scientists realized, well, Janssen might be the best target for the lasertinib because we can replace the Tagrisso with our lasertinib. So we do the exactly same kind of preclinical study and approach to the J&J Innovation China, Asia Pacific, Shanghai, and keep communicating and what could be the best way to prepare in terms of the in vitro and toxicity data and etc. Keep communicating such things and such meetings and teleconference was keep going on more than 2 years, and finally we can get the final destination, the agreement.
Yeah, so Tanjin, you cover all the details, good memory. So actually, I think I want to add a little bit more. So here, the Innovation Center team in Shanghai, we engaged with Yuhan back to 2016. That is actually the unique advantage of being locally in the region. So instead of asking the innovator to come to the central office in New Jersey, we have the local office in Shanghai, in Seoul, that we can be approached very easily. So our team started the discussion with Yuhan about the very unique opportunity and then socialize with our internal oncology leaders. So after 2 years for the discussion, we finally made the deal. So I would say, first of all, we came together because we have the common understanding on the unmet needs. So lung cancer is one of the most challenging cancer in the world, especially in Asia-Pac, that are accounting for more than 60% of the lung cancer-induced mortality. And also, it is a very complex disease. So usually when the patient is diagnosed, it's already at the advanced stage. So there's a huge need to address the early detection, diagnosis, and treatment. I think also another point is, we really feel we have the shared value with Yuhan about the science, innovation, and how we want to collaborate together. It's not a win and lose discussion along the whole process. It's really each party think how we can make the other party more successful. I still remember very vividly back to 2018, how the two teams worked together tirelessly, having so many late night calls and online meetings, face-to-face meetings to discuss through the scientific, the clinical, and the future, pathway. So very happy we made the deal. And I think our partnership will be continue, right, as we see more and more exciting data coming from the combo therapy.
Yeah, I think that's a really important point that you made, that this wasn't just, viewed as a win-lose or, handing off the technology to the large pharma partner and expecting them to understand it, frankly, as well as you do, or to, bring the same kind of passion and devotion to its development that you on the smaller partner had to this date. So, Taijin, could you maybe comment on sort of what you all have done to ensure sort of the continued success? How have you stayed involved in the development of lazertinib at Yuhan, and give us your thoughts on why you think that's been a positive and how that's been a positive thing for you.
Yeah. So under the original agreement, all the development, including the mono and combo, should have to be controlled by the Janssen. But we can fully understood because of the internal strategy and overall a global condition in the competitive landscape compared with the other standard of care and developed country, Janssen decided not directly develop the monotherapy. So in that decision, after that decision, Yuhan top management kept discussing regarding those situations with through the JSC, and finally we decided that Yuhan can develop the monotherapy by ourselves. So that kind of enthusiasm to build up the value, whatever at the end of the day, Yuhan keep working on the global phase 3 clinical trial, first-line monotherapy, and the result looks really good. We presented that ESMO Asia last December. So that maker, even though it doesn't cover in the original contract, but after we amendment and change a little bit differently, and we can contribute those development. And
finally, that really gave us more value, and not only the mono, but also the combo therapy. So that is the relationship between the Yuhan and Janssen. And also one more thing is in that overall development, there was a top management CEO change in between the development period. But original CEO has a really heavy enthusiasm to develop this, and the successor CEO, current CEO, also have that kind of passion. So fully support whole clinical development team and wanted to really make a blockbuster drug in the near future. So all kind of, even though original, I mentioned one more time, you guys have to have that kind of flexibility and keep believing the other parties and try to put more effort on this more value of that specific asset. Then finally we can have a more experience through that global phase 3 clinical trial, and also a strong relationship between the Yuhan and Janssen, because Yuhan fully support the clinical strategy and etc.
Yeah, Don, maybe you have some thoughts to add to that. I mean, the importance of, as Taijin said, the importance of flexibility, and Mike's comment earlier about being able to pivot doesn't just extend to capital raising and, you know, sort of company strategies, but it is an important component of collaborations as well. And maybe you could share a few more thoughts on sort of where those pivot points were here in the Yuhan, lazertinib, Janssen collaboration.
Yeah, so the priority for us in the partnership is to make sure our partner does well. So the biotech company, always make things very quickly. We don't want to slow things down. So actually, Janssen, we made the accelerate the pace. That is not quite normal for large companies in the world. And also, we focus on lining up the incentives with our partner to make sure there's a right efforts, right investment in the place. I think what make the deal so valuable for Janssen, for Yuhan, is so we both parties come together to contribute our strengths, that we could do something more exciting that none of us could have done alone. Janssen has an integrated strategy to change the trajectory of lung cancer, to change the treatment paradigm in the EGFR mutation pathway. So we focus on this critical pathway from multiple angles. So while lazertinib, they have the unique feature to target the EGFR receptor inside the cell. So Rybrevant from Janssen, we target both EGFR and MET on the tumor surface to target both receptors. So the collaboration with Yuhan allow us to reimagine the potential of the combination therapy, that we can bring something more potent, more novel to the patients.
Okay, we're running, short on time, so I have one final question for each of you, and then we'll see if we have a few minutes, 2 minutes for some Q&A questions as well. But, and this is not ending on a negative, but it's an important, with each collaboration and partnership that you do, to learn from them, and to consider what lessons you've learned and how you apply those positively going forward. So maybe each of you could comment briefly on some lessons that have learned from this relationship and how you turn them into future successes with other partners, with other APAC partners down, and what you all did, Taijin, to turn those into successes with, the subsequent deals you did with Boehringer Ingelheim and Gilead and some others.
So actually, we learned a lot. So it's hard to say, one or two things we learned from the Yuhan collaboration. But very generally speaking, as a Asia-Pac Innovation Center, we have been working in the region for 8 years. So we got into discussion with many, many different partners. I see a few key things to make the partnership successful. So number one is when you talk to a company, large corporation with J&J, it's critical to understand the priority and align the goal and the management with a potential partner. And also to really be open from the very beginning to think how to maximize the success by combining the strengths, the expertise from both parties. And I think as Tianjin mentioned earlier, along the collaboration, we also need to be prepared to be flexible, to adjust the plan. And also it's not signing the deal, it's the end of the whole thing, right? It's just at the beginning. The two teams need to work together even more closely after that. So really think about the long term, how you really want to drive the asset development by leveraging the two parties' collaboration.
Thank you.
So regarding this, after the whole agreement and amendment and all the development, we feel what we have to understand and keep in mind is you don't get what you deserved without a negotiation. So especially when you do the discussion for the agreement, if you don't have a room to discuss or some point you can give for other company, then it's really tough deal and hard to make a complete agreement. And the other point of view, you should have to know what you need, what you want, and reversely, what other company want and needs. So those two cannot be dovetailed at all. So sometimes you have to keep discussing, and if their needed is really reasonable, and you can a little bit change your strategy and keep doing for the value of the overall asset. Then might be the what we run from the overall collaboration through this razor team.
And as your lawyer, I can say your willingness to consider creative solutions is extremely important. Just briefly, I think we have time for 2 questions. If anybody has anything from the audience for our guests. Okay, well, if not, if you could, Mike, go ahead.
So there was a discussion at J&J of opening an innovation office in Seoul.
So we have our presence in Seoul. Yeah, actually, we collaborate with Seoul BioHub, the KD Seoul Metropolitan Government to initiate the Quick Fire Challenge for 6 years already. And we have 12 awardees so far. So very happy, to tap into the ecosystem more deeply. Thank you.
Okay, if everybody could join me in thanking Dan and Tianjin, please.
So thanks for sharing the inside story. I guess the key point in collaboration is, after all, teaming up with your partners.
Today, really have enjoyed the program so far, and BioCentury is very happy to support this event with KoreaBio and Sidley. So let's get started on our second presenting company track. Our first company presenting on this track is AffaMed Therapeutics, and presenting for AffaMed is Vijay Karwal, the CFO.
Thank you, Josh, and thank you to BioCentury, KoreaBio, and Sidley for giving us an opportunity to introduce AffaMed to you. AffaMed Therapeutics is a China-headquartered specialty therapeutics company targeting the development of a global platform to both develop and commercialize treatment solutions in 2 distinct therapy areas: ophthalmology, and CNS. We've been in existence for approximately 4 years and are proud to have achieved what we think is a strong track record of progress during that time. Let me share a couple of highlights about our company with you on this slide. As mentioned, we have a dedicated focus on developing treatment solutions, and I use that word intentionally because we look at treatment from a disease holistic perspective and are open-minded about using different modalities in creating optimal solutions targeting disorders, as mentioned, in ophthalmology and CNS. We are a global company. We have close to 100 team members, principally in China, but also based here in the United States. We have a full set of capabilities from in-house preclinical development capabilities to clinical and more recently commercial presence as well, with a particular and more proprietary unique capability in the development of digital therapeutics. We are developing IP both for the China market, but more recently also for the global markets, and have a demonstrated track record of development. By this year, we anticipate either conducting or having completed 12 clinical trials, with additional ones to be added, and that is leading to genuine outcomes. In the next 2 years, in particular, we anticipate filing up to 4 NDA or marketing authorization filings in the major commercial target market for us, which is China, but potentially having out-licensing opportunities for global markets in that period as well. We already are a commercial company in some of the ancillary markets of Greater China, specifically Hong Kong, Macau, Taiwan, and hope to be adding Singapore to that list this year as well, where we market 15 SKUs across 5 unique products at this point in time. I think we have been one of the most prolific companies in terms of BD and partnership, reflecting on the topic of the panel that we just listened to. As a company, we this week signed our 11th partnership for a total of 19 programs within our company. And those partnerships are all international, and I think represent a strong validation for the company and its capabilities. And most notably, this week, having signed our first partnership with a big pharma, Eli Lilly, we are very proud of the validation that we think that represents. Our strategy in a nutshell, or perhaps 3 nutshells. Firstly, we are principally focused on near-term commercial launch opportunities in the China market, the second largest healthcare and pharmaceutical market opportunity in the world. Secondly, as mentioned, we are increasingly pivoting to developing and using our capabilities to develop IP and value that can be monetized in global markets as well. And then thirdly, the pathways through which we build value along those 2 vectors are firstly continuing to use our BD to build our commercial portfolio in China, and secondly, using that same capability in the other direction by bringing monetizing the development for global markets. And one way of thinking about that is what we call global to China, but also China to global. Let me share the highlights of our portfolio. As I mentioned, we have quite a broad portfolio of 19 programs, but within that, we identify 3 principal value drivers. The first bucket is what we call near-term launch opportunities in neurology. We signed this week during JP Morgan an agreement with Eli Lilly, in which they have given AffaMed the rights to launch their CGRP, which we believe is a best-in-class in this category, marketed in the United States under the brand name Emgality. INN is galcanezumab. This is a very near-term opportunity. The NDA has already been filed in China, and AffaMed anticipates launching this as early as the 4th quarter of this year. We also, in neurology and targeting actually a similar target audience from a physician perspective, have a digital therapeutic that is close to market. This is a first ever treatment for cognitive impairment following stroke, and that too is on a relatively similar timeline. Our second bucket of opportunities is what we call near-term launches in the ophthalmology space. This is principally in the surgical area. China has arguably in both percentage penetration and in absolute numbers the greatest unmet need globally in terms of cataracts. This is something that the Chinese government is acutely aware of, and they are strongly promoting growth in treatments, which translates in very significant growth in procedures, which may total as many as 10 million a year by the end of this decade, far eclipsing what happens in any other market. We offer an integrated treatment solution in this area. We have both best-in-class advanced intraocular lenses using extended depth of focus technology, and we bundle those with a pharmaceutical that optimizes post-surgical care for patients receiving cataract surgery, both by reducing the risk of inflammation as well as pain. For both of these, we expect to file NDAs, or in the case of the device, application for marketing authorization within the next 12 to 18 months, and then hope to be a commercial presence in China in these products by the end of 24 or early 25. And then the third bucket is more R&D driven. These are what we believe to be best or in potentially first-in-class opportunities for the treatment of retinal disease. In the wet AMD space, we have a bispecific antibody targeting VEGF and Ang2, a mechanism similar to the Roche's Vabysmo launch, but a different molecule. We believe potentially a superior molecule. We have global rights to this product, a very significant potential economic opportunity. We will hopefully be able to announce interim first human data as early as this summer from a trial that we anticipate completing in the early part of next year. And we also, in partnership with a US-based innovator called Allegro, and actually a Korean partner, topical for today, called Hanmi, for what is the first compound to have ever demonstrated the ability to reverse disease progression in dry AMD. Dry AMD, as some of you may know, is an area that is attracting a lot of attention. Well-known companies like AbTis and Iveric here in the United States are close to approval in that area. The market has rewarded them with that, with cumulative market cap of probably close to 10 billion. And that is for products that slow disease progression. Our molecule can actually reverse, or at least we believe it has the ability to reverse disease progression. Very briefly on our partnership with Lilly, migraine affects 1 in every 10 Chinese. The rate of patients presenting themselves and then subsequently being accurately diagnosed is far below what we see in the United States. So even with those lower rates based on China's large patient potential, today there is a market opportunity that is equal or already exceeds the United States. Research analysts expect this molecule to surpass $1 billion in annual sales in the US next year, which I think is an interesting indicator of the potential that this may hold in China. Interestingly, leveraging our digital capabilities, what AffaMed will do in conjunction with the launch of this product in China is also launch a digital companion, which was developed in-house in partnership with academic partners in China. This is a tool that will serve both as a tool for physicians to optimize diagnosis and possibly also as a disease management tool for patients. And we think that particularly in an indication that is identified, underdiagnosed, like in China, that this will be a critical and very strategic tool that we can bring to bear in growing the market opportunity for this product. All of this over the next 12 to 18 months will generate a significant number of pivotal milestones between trial readouts, NDA filings, as well as commercial launches. We, like I think many others in this market, anticipate using these milestones to raise additional funding. In closing, to date, as a company, to support all of the significant work that I've been pleased to present to you today, Our shareholders have supported us until now with a little over $250 million in funding, including our principal shareholder represented in the audience, CVC Group, but with additional backing from Lake Blue Capital, Partners Investment, a well-known Korean VC, and a number of other hedge funds and family offices here in North America. So with that, I'd like to conclude my remarks. I know I've probably gone over time, knowing myself.
That's okay.
But always happy to take a question. Thank you.
Vijay, thank you. Question from our panel of investors.
Yeah, so I have one question. So you are expecting the commercial launching in this year in China.
Yes.
So are you going to sell by yourself or trying to find a marketing and sales partner?
We are already in the process of building out our commercial capabilities. The benefit we have, and I'll keep it brief, is that in the two areas where we are closest to market, which is the solutions for cataract surgery as well as migraine, that patient concentrations are actually quite high in China. There is approximately 300 hospitals that treat, as close to 40% of all migraine patients in China. So you can imagine, it's a manageable effort from a sales perspective. In cataract, as you may know, eye surgery is one of the few areas where the private sector is very dominant in delivering treatment in China. And it actually means that, frankly, a relatively limited number of strategic partnerships. The most important player of this is a chain called Eye Hospital. AffaMed is already working with Eye and treating patients on a pilot basis in some of their hospitals. So the nature of these target markets actually allows even a younger company like ourselves to have a very credible pathway towards commercialization.
Hi, nice talk. I like the use of the digital companion. I'm just wondering how you're positioning it. Is it as part of the approved therapeutic or just optional extra you're adding one?
It's it's a separate product. To be honest with you, we see potential even for it as a standalone revenue opportunity. But I think in the first instance, I think our principal objective is to grow our target market for the migraine therapeutic as quickly as possible. So that's the way we're using it. And to give you, an anecdote on that, over this past summer, we tested this digital companion in about 1,000 patient visits across 30 hospitals in China. Two of the principal outcomes were that relative to the baseline, we saw accurate diagnosis increase from 75% without our diagnostic to over 95% with our diagnostic, and possibly most important, the time that the patient spent with the physician was reduced by about 25%. And time is money for every doctor, but particularly in China, that's an even greater incentive. So we think this is potentially a very powerful tool.
Thank you, Vijay. Much appreciated.
Pleasure.
So we're now going to have our second company presenting on the track. Many of you know Insilico Medicine, and presenting for Insilico is the CBO, Michelle Chen.
Good afternoon. I want to thank the organizer and the second presenting company for your flexibility to have another meeting at a run. So it's my great pleasure to introduce you in Insilico Medicine. We're an AI-driven drug discovery development company. Insilico Medicine was founded in 2014 in the United States, started as an AI company. Today, we have grown to about 220, 250 employees worldwide. 40% employees focus on AI, machine learning, and the other 60% focus on drug discovery development. We're headquarters is based in Hong Kong, but we also have global offices in New York, Montreal, UAE, Taiwan, and Shanghai and Suzhou. So what claim to fame in Insilico Medicine is the pharma AI platform. What differentiates this AI platform from the others is kind of end-to-end solution. We focus on three bottlenecks we see in drug discovery development. The first bottleneck is biology, where a lot of novel drug discovery, target discovery has been challenging in biomarker discovery. The second bottleneck is chemistry. How do you come up with using AI to come up with novel ways to design small molecules? It's been very challenging in this field. The third one, we all know that clinical development is the most time-consuming, costly part of a drug discovery development. we have figured out a way, AI tool, where you can predict clinical trial success, particularly with transition from phase 2 to phase 3. This comes very handy for investors, for business development, as well as the clinicians. So based on the Pharma AI platform, we'll be able to build about 30 programs in the internal pipeline with focus on fibrosis, oncology, inflammation. We also have a COVID program. On top of that, we are working with many global pharma companies. About 50% top 20 global companies are using our tools or in some set of collaboration with us. Last but not least, we're well published in this field, about 160 peer-reviewed publications, with about $400 million in to advance molecules as our partnership. So here's a snapshot of the management team. Alex Zavankov is the founder CEO, and together with Alex Alper, our president, both of them have an AI background, formed the company about 8 years ago. We have our CSO, now as co-CEO, Dr. Ren. I'm from big pharma, a lot of drug discovery experience. Myself, a mixed background in pharma, biotech, and I'm based here in the Bay Area. Our CFO, Nirav, from Wall Street. Recently, we hired Dr. Sujata, who heads our clinical development. Aside from the experienced management team, we have a team of very world experts in biology, chemistry, and AI who can help us to design paths forward for the company growth. Very luckily, we're supported by well-known investors. You may recognize some of the names here. Very interesting about the investor mix is that really we have tech supporters such as Baidu, Innovation, and Bolt Capital. We also have more traditional biotech investors such as Qiming, OrbiMed, Lilly Asia Venture, as well as financial investors such as Warburg. So there are 3 pillars that build upon our Pharma AI platform. The first one is Pandomics, basically a collection of omics data, text mining capability, where you pretty much scroll about close to 2 trillion data points with about 40 in-house curators, collect all the information, and use this very powerful tool. You can identify novel targets, and looking for biomarkers can be used for translational medicine. So this tool is widely used by many, many customers today. The second one is very important, our kind of pillar of this whole Pharma AI is Chemistry 42. It uses machine learning approach as well as GAN, which stands for Generated Adversarial Network, and to using AI to imagine small molecule folding. And this is based on a training set of over 1 billion molecules that with experimental data. Today, many of the pharma companies, larger pharma companies are using this tool. Thirdly, we just launched in clinical is, again, machine learning approach, and we can predict clinical trial success or failure based on the design, the target class, admin profile, and other factors. Again, comes very handy from investors as well as BD as clinical development. Very recently, we launched the 6th generation AI/automation. What means that we can take automation, automated labs we're currently building in Suzhou. If information come from high throughput screening, high content screening, next-gen sequencing, and other imaging systems, combined with the AI modules such as Pandomics, Chemistry 42, and others. And altogether, we can make biology more predictable. Again, our whole idea here is to advance discovery work and shorten the timeline, reduce the risk. I want to kind of showcase, based on the Pharma AI platform, we'll be able to validate, we'll be able to quickly come up with 10 preclinical candidates in the last 2 years. Our most advanced asset, we just announced completion of phase 2 study, entering phase 1 study, entering phase 2, where it took us less than 2.5 years to get to the phase 1 stage from the beginning. We also have suite of assets built upon Pharma AI platform. Here's a couple examples. So aside from target X, we had PhD2, QPCTL, USP1 currently entering IND filing stage. We expect to enter in phase 1 and in the first half of the year. Many of them are available for partnering. So we're very fortunate to have a lot of partners working with us. On the one hand, you have platform users, those are software subscribers. On the other hand, we have strategic collaborators. Recently announced a deal with Sanofi with $1.2 billion buy box and 6 targets where we'll be making small molecule for their nominated targets. Our business model is very flexible. On one hand, we have software tools you can use, you can play with to empower you to do your own drug discovery. On the other hand, we have sweet assets available for partnering, for licensing. In the midst of where we can work with pharma companies, be part of your external R&D, building assets for your pipeline. This is my last slide, and to showcase we're here to partnering, to talking to investors, engage us. We truly believe if the power of AI can really accelerate drug discovery development. Thank you.
Great. Thank you so much, Michelle. We're going to have questions from our investor panel now.
Yeah, very impressive. Just one question. I know you guys are so a lot of AI drug discovery companies, because they have a lot of assets, they tend to spin off or they do a joint venture with other companies. It seems like you guys have a manpower to do, R&D within yourself. And I know it's going to take a lot of capital to develop all those like fancy pipelines. What's your strategy in terms of the capital allocation?
Yeah, excellent question. I think internally we're going a couple of things. One is portfolio assessment, trying to prioritize those assets, right? Because as we move further to the development, the cost increase significantly. Secondly, this year, top order for business development is asset out-licensing. So we're actively talking to global partners as well as regional partners to discuss licensing assets. Thirdly, we have investor VC approaching us. They're very interested in the new co-formation. So some of their assets are giving them a jump start. So if any of you are interested in that, we'll be happy to discuss.
Hi, Michelle. Thank you. Question. It's an end-to-end solution from drug discovery to optimization and clinical. What are the other missing parts that you would need within that process? For example, you would eventually you would rely on CROs in the early stage and go into clinical for your internal pipeline. Any missing gaps that you would need in the near to midterm to become fully AI automated and, for lack of better words, reduce reliance on CROs in terms of a time?
Yeah, I think that's a very good question. So for CRO model, we are being capital efficient. We're currently working 40 plus CROs. Majority of them are in China, some are in Europe and US. We also provide a lot of redundancy in the CRO. If there is a COVID outbreak in China, CRO cannot function, and we can find other regions CRO can. So there's a lot of where all the machine CRO network we have been working with already. In terms of other things we can do better, I mean, last Wednesday we started as an AI automated laboratory. The idea here, we all know chemistry is more predictable than biology. And in the mind of a computer scientist, right, the future is predictable. It's a matter of probability, right? How can we make biology more predictable and what can be used for drug discovery? And AI can collect all the information, not only just from high throughput screening, but high content screening, so that we can build new modules for AI and predict if put this molecule, what else may happen, and what about other recommendations for drug repurposing, right? So AI start thinking along the same side where human will be thinking. We'll complement some of the blind spot we have, but hopefully offer new insights.
Great. Thank you so much, Michelle. I'm afraid we're out of time, but if other folks have questions, I'm sure Michelle would be happy to speak with them. So thank you.
My colleagues will be around, so feel free to talk to me.
Great. Thank you.
So thank you so much for giving opportunity to present today. We are the stem cell global leader at SCM Lifescience, and this is our building. It's great outfit. So I want to introduce. We have two important people in our company. The founder, Dr. Song, and the CEO, Dr. Song. And he's developed all of process the stem cell medicine product, and he is focused on the first-in-class medicine product. And our company is to provide high quality, thank you, high quality mesenchymal stem cell medicine in the worldwide, and that we are trying to do, support the intellectual disease patient also. And would like to introduce the core technology. We are the mesenchymal stem cell product company, and the advantage of the mesenchymal stem cell has the multiple source and the large scale production and multiple lineage differentiation, as well as the immunomodulatory and the anti-inflammatory function. That is very important for the the tissue the, dysfunction or the increase their tissue function or regenerative kind of that. And there are three different type of the patent about the mesenchymal stem cells, the isolation and differentiation medicine. Well, only the two companies has the isolation, the patent. The first one is a density graded centrifugal method. We call the GCM. The other one is SCM. Ours is a fractionation, sub-fractionation culturing method. Before they introduced our technology, the previous one, the GCM, has a little bit limitation because after centrifusion, and we tried to get a single layer, and this is expansion the cell, but the mesenchymal stem cell, that layer only not only the mesenchymal stem cell, but also other cell type also is 5% more. So that the other cell types make a problem about the low purity and the low efficacy. There is published to the New England Journal of Medicine and the Nature Biotech. They support, they point out about these 2 points, and this might be making the result about the clinical trial failure or the denied from the FDA. Okay, so this is our technology, sub-fractionation culturing method. So just bone marrow mixed with the culturing media, and then the transfer 2 times only the supernatant, and then start making the colony only from the single cell and then making the colony. And then we just collect the colony isolation and the expansion, and then making finally the library. And this is a great, better the cell type compared to the GCMs, the high cell density and the formation of the colony, and then the better efficacy. So our SCM is, compared to the GCM, our SCM technology is showing the high purity mesenchymal stem cell, the high efficacy and the high efficiency, as well as we have the core technology. So after making the library, we have the own technology about the selection of the disease-specific mesenchymal stem cell based on the MOA, and then showing the in vivo, in vitro, the efficacy test. And so finally, it depends on what kind of the indication. And we have the AT patent and then PFT publication, and we have the pipeline, about the 3 different pipeline. But about our product is 2 different formulations. The first one is fresh and the frozen, very easy. The fresh one is a little bit the early stage. We developed the fresh one, but now we have all the process is by the frozen. Of course, the fresh is for one of our clinical trial is try to use the fresh one. Anyway, we have the 3 different pipelines: atopic dermatitis, acute pancreatitis, chronic GVHD. Because mesenchymal stem cell can do the immunomodulation function, they have the immunomodulation function. So we selected the 3 different diseases about the anti-inflammatory relative diseases, and all of them. This is a recently update about the patient. We already recruited all of the patients, and the atopic dermatitis case, we will get the clinical trial, clinical report. We will get this coming May. And the acute pancreatitis last August, we got the page 2A the result, and the chronic GVHD by the end of this year, we get the data. We want to finger cross. Anyway, the GVHD case, after hematopoietic stem cell transplantation, patient start to the immune reaction, and then some tissues has something problem, including inflammation or a kind of dysfunction. And we have a MOA, and depend on the MOA, we have the selection of the mesenchymal stem cell for GVHD. By the cell surface marker, and then in vitro efficacy test, and the in vivo efficacy test also. And this is a therapeutic use. We have the data. And we did, for the therapeutic use, we have 3 patients, GI tract, the liver, and the mouse lung GVHD. GI tract patient case, before treatment, their tissue is worse. But after injection, then 10 days later, start the improvement, and then after 3 months later, it looks like back to the normal. And the liver also, after injection, their liver, the bilirubin, the level is going down, and the second injection also going to back to the normal. and then the mouse also the severe to the mild. That is a great performance by our domestic stem product. And the acute pancreatitis, about the heavy alcohol, like gasoline is the main region. And then after injury, the acinar cell in the pancreas, there is lesion to their protease. So it means that degraded other cell type also. That is start to the cytokine storm and then making the inflammatory condition of the pancreas. And this also still no drug about the treatment of the acute pancreatitis. So if ours is a nice efficacy, that is a first in class. And there is we have a published and we have MOA. The best on this one, we selected mesenchymal stem cell for the pancreatitis, and also we last August we got the clinical data about the phase 1 and 2A. There is no safety issue. We confirmed the safety, and efficacy test also is very nice. Very nice data we got that from the CRP level and the IS6 level. That is for the pro-inflammatory factors, and also other two, the modified Marshall score and the CTSI. That there are both them is physical testing, the physical monitoring about that. So these two score also two score index is going down. So we confirm the efficacy also. The atopic dermatitis case, there are lots of the target by the many of the company, but we are trying to do, focus on the just immune modulation system in our body. So we have the MOA and then the select these cells and then to try to do the therapy to use. So after treatment, our mesenchymal stem cell product, after 12 weeks later, is gradually improvement of the skin problem. It's a terrible skin, but it's gradually better. And then the phase 1 study showing the low dose and high doses, showing the increased the therapeutic effect. And then at 12 weeks and 24 weeks, we got almost concludes the 90% above. We got the nice data about the EG score, EG50 score. And there is no safety issue so far. And then this is the strategy for the phase 2 study. And then this is the milestone for phase study and the clinical study report we will get on this coming May. So that is, we are very excited about that. So it depends on if that confirm, might be our company's value might be increased. And our future plan about the clinical trial, the timeline is atopic dermatitis case. We will do after May, we will prepare for the phase 3. And that is, we will working with the Handog, our partner in the South Korea partner, Handog, we will do the phase 3. And acute pancreatitis, now we are trying to prepare the phase 2B. And then we will do the 2025, we will do the conditional approval. That is, we are expected. And also chronic GVHD case, end of this year might be finished over our phase 2 study. And then after that, the 24, we will do the conditional approval. We are prepared about this one. Also, other two, the pipeline is still the research grade.
Okay, I think we wanted to save a couple of minutes for questions if we could.
Yep, this is the one of the tissue engineering project. And then we have also another strategy for our mesenchymal stem cell has now is the first generation. The second is a priming condition by some cytokines, and the third one is a gene editing system, which is effective the genes editing. And this is now second generation, the priming. Also, we got the nice data about the rheumatoid arthritis, and now we are the scale-up process developing using the 3D bioreactor system. Better than almost the 6 to 7-fold increase the cell amount, and we can supply for the patient also.
Okay, I think we're gonna have to. I'm afraid we're gonna have to cut it off here. Just we're running out of time. Want to make sure. Thank you so much for the presentation, Dr. Kim. Want to make sure we have time for at least one question from our panel.
Thank you. Yeah. Thanks for the nice presentation. There are obviously more. So quick questions on the corporate strategy on the two pipelines in phase 2, atopic dermatitis and acute pancreatitis, given the phase 2 data is to be in May. Any plan in terms of a next step?
Yeah, so after get the data, actually now we are trying to find a global clinical trial partner, as well as the tech transfer or the license out. That is our current strategy for this year or next year. That is ours. Yeah.
Great, thank you so much.
Yeah.
Very much
Thank you.
appreciate the presentation. So our next presenting company is Profound Bio, and presenting for the Profound Bio is Baiteng Zhao, co-founder, chairman, and CEO.
Okay, first of all, thanks for the organizer for the opportunity to introduce Profound Bio here. My name is Baiteng Zhao. I'm the CEO of this company. Profound Bio is a clinical stage company focused on development of next generation ADC therapeutics for cancer patients using our novel proprietary ADC technology platform. We are located in both Seattle, Washington, as well as Suzhou, China. So over the past 3 years, we have raised nearly $140 million from these well-known investors. So our leadership team has accumulated many years of drug development experience from both large pharma and smaller biotech, especially in the area of ADC therapeutics, from discovery to clinical, from CMC to regulatory strategies. So our pipeline are mostly focused on solid tumor targets with both first or best-in-class potential. Our lead programs have been cleared by US FDA for first-in-human study, and more ADC will come to the clinic later this year or by early next year. So ADC is a complex kind of molecule with 3 major components: antibody, linker, payload. So the primary mechanism of action is through binding to the cell surface antigen, internalize, release a small molecule, and kill the tumor cell. And the goal is really to expand the therapeutic window of those small molecules. The wider you can expand it, the better ADC can be. So there are already a dozen also ADC approved, but there's still so many failures. So there are still a lot of room for improvement, especially for ADCs using a very hydrophobic payload. The reason is for solid tumor, we want to use a hydrophobic payload in order to have bystander effect to improve our efficacy. At the same time, because of those hydrophobicity, the physicochemical property of the ADC is suboptimal, so it affects its PK/PD. So we need to improve the physicochemical property of ADC so that I have a better PK/PD and basically a wider therapeutic window. And our approach is really, once you have determined on the antibody and the payload, you work on try to optimize through the linker component. So we try to use the linker to shield the hydrophobicity of the payload to improve the ADC. So our linker uses the proven technology, let's say conjugation to antibody through the cysteine conjugation, and also cleavage through the typical peptide cleavage. And our drug linker can conjugate from anywhere from 4 to 8 payload per antibody. Just the bottom 2 are just the structure of our current drug linker. To your left is a STK payload. It's a topo inhibitor. To your right is MMAE, it's a tubulin binder. The structure will be disclosed or published very soon, so you will see the structure very soon. This is just a quick snapshot of our data. So basically, on the top, it shows how our technology can improve upon the current available technology, how on the hydrophilicity, and then it's PK and PD. So so the black curve, the black curve is the antibody to look at the hydrophobicity, and then the blue one is conjugated through for a drug load for the ADC. And then compared to the red one, which is the without conjugated ADC, we can see a much greater improvement in hydrophilicity. And with that improvement to our lower left is the PK profile. So our ADC with the same payload, but with twice as high the DAR value, the conjugation value, we can still have a better PK profile and comparable to the unconjugated parent antibody. And to our right is the PD CDS model, where it shows with half or a quarter of the dose, it still outperform the technology, the current the vedotin technology. Vedotin technology actually has been used in 4 of the 14 approved ADC products. And another quick snapshot of our technology through comparing different HER2-based ADC. So the blue curve is ADC using our technology, and the green one is using the technology from Daiichi Sankyo, and the red one is the TDM1 from Genentech, and the purple one is using vedotin. So from left to the right is 3 different cell line models with lower and lower target expression level, HER2 expression level. So you can see ours perform really well in this model, especially at the medium to low expression cell models. And just the last 2 slides show you quickly our lead program. The first one is 1184. It's a folate receptor alpha targeted ADC. This target has been validated with recent approval from Imagin. And based on the preclinical data, we use a different payload with different mechanism of action. We believe there's a room for us to greatly improve the clinical efficacy and safety profile. So this one has been proved, for first in human study, and has been first patient has been dosed last month. And the second program is CD70 targeted ADC. Again, this is ADC IND has been cleared by FDA, so it's ready to start to recruit patients in a few months. And this target highly expressed in renal cell carcinoma and Hodgkin and non-Hodgkin lymphomas.
Great. Thank you so much, Dr. Zhao. We have time for a couple of questions from our investors.
Yeah, thank you for your presentation, especially some how-to-load data is really, really impressive, actually. And then, so I want to know about your one of your slides. You told us that you're going to have a clinical trial from this year, and then I want to know they're in China or clinical study in the United States.
Yes, both programs have been approved by U.S. FDA for first-in-human trials. So we are starting to trial in the United States right now.
And if so, and then what is your strategy for the later on? So are you going to seek partner like a big pharma later on, or just develop by yourself?
Yeah, as a startup, these are our first 2 programs. We have many programs in the pipeline. So for our lead programs, definitely we are very open to partnership, so we can accelerate the development of these therapeutics for patients.
Yeah, okay. One last question is, so how about some IP status about your novel linker?
Okay, so the entire pipeline is we have global rights, so we develop antibody ourselves. The drug linker technology with different payload, different drug structure is all developed ourselves. So it's our internal technology, and So IP have been filed, and like I mentioned, the structure will be disclosed or published very shortly. So that means it was applied in 2020. Yeah.
Thank you very much, Dr. Zhao. So I know the ADC market is so competitive right now, right? There are many, many different players. So what will be your specific strategy, how you can speed up the timeline and to win the future market? Maybe you can share a little bit from the clinical development, regulatory perspective.
Right. From the very beginning, when we start a program, we have to think in the at the end game, right? TPP or target profile. So basically, we have to choose a target for the indication for the population with unmet need. So with that, and also look at the potential competitors in the next few years in both clinical development and in commercial. So we believe what the first two, at least the first two program, disclosed program, we have, at least for the first one, we believe we have best-in-class potential. And for the indication we are going after, there's a huge unmet need. So the development path can be relatively straightforward. And for second one, it actually can be a first-in-class ADC molecule. And then for indications like nasopharyngeal and renal cell, I think those are also with a high unmet need. And that's coming to a question. That's part of the reason we haven't been choosing heme-targeted ADCs. Yeah, so that's part of the strategy.
Great. Thank you so much, Dr. Zhao, for the presentation. So we have one more presentation on this track, and I'm happy to introduce Abion. And presenting for Abion is Jake Yeo, the IP manager.
Hi, nice to meet you. I think I'm the last turn, and there are not many people left. I think I should do it fast as fast as I can. I'm Jake from the Abion. I'm the IPR team leader of the Abion. And in this point, I think about my parents. My parents always said most valuable things always at the end. So I hope our Abion IR will be a great value to your investors. So our Abion is founded by the Yang Shin. He's a professor of the Seoul National University. He established Abion in 2007 with the concept of precision oncology. As you know, precision oncology in 2007 is a very pioneer idea. So not many people understand what is the concept and why do we trying to do this. And in 2019, we start the Abion 401 as a precision oncology drug. So we start the clinical trial, and 2021, we finished the phase 1 clinical trial, and we listed on Kosdaq. So recently, our CEO always say like combining the Adidas, marketing mention and the Nike marketing mention. So just do it, and impossible is nothing to all his students and employees. So we are listed in Kosdaq, and we start a phase 2 clinical trial in 2022. And it's ongoing right now. So as you can see here, we have a very lots of pipelines. Actually, we have 12 pipelines, and our employees are only 35. So we are always on the crunch mode. But our lead pipeline is on Abion 401. It's the c-Met TKI inhibitor. It targets the lung cancer patients, and we are studying the single exon 14 skipping single treatment. In this case, our goal is to finish our phase. Our goal is to report our interim report within this year, and we are hoping we can start the combination therapy with the EGFR TKI in this year. And other oncology pipeline is Abion 501, and the other one is the Abion 101. I can briefly introduce you about this. So this is the upcoming milestones, and in case of Abion 401, the phase 2 clinical cohort is start. So we are expecting the interim readout in 2023, And there's a phase 2 cohort 2, which was the EGFR TKI, the combination therapy, is planned in the first quarter of this year. And our goal is to first patient in this year. And in case of Abion 501, we are complete our USP process development, and we are starting to develop with the downstream and the formulation development. And also we plan finish the GLP tox study within this year and submit IND in this year. In case of Abion 101 is quite special to us, so we plan to complete GLP tox study in the first half of this year and complete IND submission in this year. So this is the Abion 401. Many people are very familiar with the EGFR TKI because in the lung cancer, Tagris is one of the biggest blockbuster drug, and they target the EGFR mutation of the non-small cell lung cancer. But after the Tagrisso, when the patients progress their cancer, then they have a c-MET mutation. So there are many mutations, of course, but c-MET mutation is 15% to the 50%. So it's a very large population of the second line, the third line of EGFR treatment. So it's one of the biggest markets. So our Abion 401 erased the structure of the lung kidney toxicity, and also it's a highly selective c-MET TKI. It is the most highly selective TKI in the market. And also, we have a very relatively high unbound fraction, which means systemically, it lowers the drug toxicity in the body. So we can show you the very superior safety profile in the next slide. Here, we finished the phase 1 clinical trial, and we can show you we have a great superior safety profile here. So not like other TKI. Mostly the TKI is like a 10% to 20% of adverse event, but in case of Abion 401, we have 0% of adverse events. So which means there's no patient who stopped the drug or who showed the grade 3 or more. And luckily, we are the all-comer patient phase 1 clinical trial. So we got the PR cases. We are 2 PR cases and 5 SD cases. So it's quite solid efficacy data. So why this grade 3 adverse event is important? Because it's very critical when we treat it with the EGFR TKI. EGFR TKI also have some kind of toxicity. So if we treated 2 drugs all together, then the adverse event is getting much more severe. So hypoalbuminemia and ILD is the very common adverse event in the c-MET TKI, and also in other drugs, there's a liver toxicity also. But Abion 401 has no toxicity in all these adverse events. So our key milestone is to, now we are in the phase 2 clinical trial, and we are talking with many global pharmaceutical companies, and many of them are discussing about licensing out or something, and they really wonder what is the efficacy data, because we have a small number of patients in the phase 1 clinical trial. So if we add more patients, and as I mentioned, we have a plan to report the interim report in 2023, so that these kind of milestones can be very accelerated. And we're aimed to finish 2 clinical trials, and also we plan to start the EGFR TKI combination therapy. We already published this kind of in vivo data in previous ESMO 2022, so you can easily search our data. And our second pipeline is Abion 501. This was the first-in-class Claudin-3 antibody. Not everyone is recently very familiar with the Claudin protein, because before 3 or 5 years ago, nobody understood what is the Claudin is. But recently, Astellas report the success of the phase 3 clinical trial of their Claudin 18.2, and also the AstraZeneca had licensed in the Claudin 18.2 antibody. So many people wondering what is the Claudin protein. So our Abion 501 is target the Claudin-3, which was the family of the Claudin protein. And this is the novel target antibody, which means There's no other antibody in the market that target the Claudin-3. Also, it has a high affinity and specificity, and not like Claudin 18.2. In case of Claudin 18.2, they target the pancreatic cancer and the GI cancer, but our Abion 501 can target various indications. So here you can see there's a breast cancer cell line and ovarian cancer cell line, colon, gastric, liver, pancreatic, and lung. So we tested our Abion 501 in the various cancer cell line, and if they overexpress the Claudin-3 protein, our antibody shows the ADCC in the cancer cell lines. So we can say that our drug can expand the indication.
Jake, we need to have time for some questions. If you can wrap it
Here,
up.
so this is the in vivo data. We tested in the breast cancer xenograft model, and our Abion 501 shows the very great data here. Also, as I mentioned, we can compare to the 18.2. 18.2 market is very already a red ocean, but our Abion 501 is exclusive, and there's no other competitors right now. So in case of, I'm going to go to the Abion 101. This was a kind of new concept. It's a broad spectrum prophylaxis antiviral drug. And this is the interferon. Many people are familiar with the interferon, and we developed the second generation of interferon, which was enhanced the stability. And this enhanced the stability makes us possible to make into a dry powder inhaler and dry powder formulation. And it enhanced the storable, and we can easily treat them to the patients who need. So as you know, the Biden administration they announced the implementation plan, and they say US need the antiviral development and manufacturing. They need at least two novel antivirus for stockpiling in 5 years. So our Abion 101 is really easy to stockpiling because we have a very stable formulation and very stable data in the room temperature. And as you can see here, we tested in the various virus like COVID-19 or the influenza and hepatitis and RSV. You know, hepatitis is not the respiratory virus, but still they are the DNA replicate virus. So we prove our concept that we can treat it to every other virus who might be infect in our respiratory system. So our key milestone will be.
We're gonna have to cut it off. We're
this is
running out of time.
Yeah.
Why don't we turn it over to the investors for a quick question?
Yeah, maybe I can start. So first of all, very much looking forward to the readout from your 401. You said this year, right?
Yes.
Yeah. So when you get the data, we'll be very happy to look at. And
oh, really? Oh, thank you very much. That's a really honor.
And also, second question for your antiviral prophylaxis. Do you have any more data that you can show?
I'm sorry, more data? This was
data.
actually the more data. Yeah. We already tested in the in vitro model, and we are expecting the monkey PK and PD data in the first quarter of this year. So after this first quarter, we can say that we complete our preclinical test, and then we'll plan to submit IND in this year. Thank you.
Great. Thank you so much for the presentation. And
very much.
Thanks to all of our presenting companies. Let's all give them a hand. And I'll turn the program over now to Jerry of KoreaBio.
So the patient ones are left. Thank you again to the aspiring 12 biotech companies and again to the investor panels. I know how much enthusiasm it takes for you to take off 3 hours or 3.5 hours off your time of JP Morgan conference. Thank you again. And it looks like we're going to see you at our 4th Global IR at the same place next year in January at the JPM week. And if you wish to share this video with your colleagues or wish to watch it again, you can do it by searching BioTV on youtube.com, and it'll be available within a week. And so KoreaBio, Sidley, Austin, and BioCentury will be hosting the same event, and I hope to see you here at JPM 2024 again. When you actually walk outside, please grab snacks. We don't want to waste it. And thank you again. We're going to end this here right now. Thank you.